Hi everyone, good morning guys, welcome back. How are you all? So guys, as you can see on the screen, yeah. So I have a very important information for all of you. Good morning, or good afternoon. So guys, this is UAITS. This is Unacademy All India Test Series just for rupee 1. So all those of you who are somehow not able to take AIMT, you have missed some, then this is for you. All of you who can, this is a takeaway series. So there are 10 minor tests involved. 10 minor tests, 6 subject wise tests, 3 major tests, and 6 overall tests with complete syllabus coverage, detailed test result analysis, 72% score correlation with NEET 2025, and you can take these tests at your convenience. Although I always believe that you should be taking these tests at the same time, that is 2 to 5 p.m., so that you set your biological clock that your brain should start working between 2 to 5. Not a very comfortable time, it is maximum sleepy time, but you have to set your clock, biological clock, for 2 to 5 p.m. So always practice these tests between this. So that is why I keep my practice classes also at 2 to 3. But yes, yesterday we could not, never mind. But our time should be 2 to 5. So take these test series just for rupee 1. Really, like, it is worth it, guys. So you can take these tests at your own convenient time though. But my strong recommendation is 2 to 5 p.m. And so just enroll it. It is a limited offer. And apart from this, guys, yes, your all India mock tests are continuing thrice a week, Sunday, Wednesday, and Friday. Right? So this is all India mock test. So next is coming up. Today is Wednesday. So you have your test between 2 to 5 p.m. Next will be on Friday and then on Sunday. Right? So guys, with this, let's come back to our NCERT reading. We have completed around 9 chapters yesterday, and today we have to complete rest of the 10. So are we all here? Please let me know. Yes, please. Are we all here? We are going to start with biomolecules. Quickly share the session with all your friends and we can start with it. Right? Shall we start? All right. Now the first point to take care of here is, see, all the elements present in the sample of Earth's crust are also present in the sample of living tissue, with few differences, and also differences in the proportion of those elements. We'll be discussing that. Now, how do you analyze the chemical composition? By making a slurry in trichloroacetic acid. See the formula also. Now, this is the solvent that we use during that. Now, when you filter it, we get two proportions. One is filtrate, or more technically the acid soluble pool, and the second is retentate, or the acid insoluble pool. Is it clear? Now see, what do you get in acid soluble pool and acid insoluble pool? You know, in acid soluble pool you get the micromolecules, and acid insoluble pool you get the macromolecules, or the polymers. But fat is a micromolecule but still found in the retentate. Why? We'll all study that. By the way, this table, guys, as I said, this table is important. See, this tells you the percentage of different elements in Earth's crust and then in the human body. I have given you the mnemonics how to remember the top 4 most abundant elements in the Earth crust and in the human body. Also see the percentages. Is it clear? Now, if I want to study the minerals present in any tissue, I use this destructive experiment. I use this destructive method. What is destructive method? We make ash out of it, because in ash, see, when we burn a tissue, what happens? All the water dehydrates, water evaporates, then all the organic compounds, they will get oxidized and be released in the form of CO2. What will be left behind? The ash will only contain the minerals. So we are destroying the tissue in order to study that. Hence the destructive method. We call it ash, and ash contains all the inorganic elements. Is that clear? Now, amino acids, coming on to that, they have they have amino group and the acidic group. Is that clear? And now their in amino acid, we call it a substituted methane. We call it a substituted methane. And we call all of them as alpha amino acids. Why? The carbon in amino acid is alpha carbon. What is alpha carbon? Chiral carbon. It has all the 4 different groups. What are the 4 different groups? Hydrogen, NH2 group, amino group, carboxyl group, the acidic group, and R group, the variable group. So all the variety that we have in the amino acid is all because of the R group, right? Hydrogen, carboxyl, amino group, and the variable group designated as R group. Is that clear? Now R group can be in variety. R group in amino, so if R is hydrogen, it is glycine. If it is methyl, it is alanine. If it is hydroxymethyl, it is serine. You have to remember these. 3 out of the 20 are shown in the figure. We'll come to that. Now, depending upon this R group, guys, depending upon this R group, if R has an additional NH2 amino group, then it becomes basic. It becomes basic here, right? If it has additional COH group, it becomes acidic. And if it doesn't have either of two, then it will be neutral. Is that clear? Then depending upon if it has another this benzene ring, it will be aromatic, and so on. So it depends upon the composition of our group that we can divide amino acids into different categories. Now see the examples. Acidic group, acidic amino acid, glutamic acid, aspartic acid, right? Basic, we have lysine, we also have arginine, which is a major component of histones, right? Neutral, in neutral we have valine, we have glycine, we have alanine also, right? Glycine, alanine, all these are neutral, while serine is alcoholic. This is alcoholic in nature, right? Then in aromatic we have tyrosine, phenylalanine, and tryptophan. Are we getting that? Now very important that it is only the amino acids which have this unique property that their both groups, NH2 as well as COH groups, are ionizable. Is that clear? Both the groups are ionizable. And see this, after getting ionized at different types, in different types of solutions, at one point of time it can be this amino group getting ionized, while in another kind of solution it is the carboxyl group getting ionized. But there can be a state of equilibrium where both of them get simultaneously ionized. Both get simultaneously ionized. Right? Now this is a situation where both the groups are simultaneously ionized and plus and minus charge coexists. This ion is known as a Zwitter ion, and this is only the property of amino acids. Are we clear till here? Now talking about the lipids, guys, water insoluble, you have to note down how many carbon atoms are present in palmitic acid. 16. Now remember that this 16 includes the carboxyl carbon also. Fatty acid, acid means it has COOH group. So if I remove this carboxyl carbon, then how many carbons will be present? 15. So read the statement carefully. Palmitic acid has 16 carbons including carboxyl or excluding. Including is the right answer. If it says excluding, then the answer should be 15 here. Similarly, arachidonic has 20 carbon atoms. Now fatty acids can be saturated. If there is no double bond, it is saturated. If there is one or more double bond, it becomes unsaturated. Now a simple lipid is glycerol. What is the chemical name for glycerol? Trihydroxypropane. And in this trihydroxypropane, there are 3 hydrogens which are replaceable by fatty acids. Is it clear? Now fatty acids, see, many lipids have both glycerol and fatty acids. So glycerol molecule will remain one, but now one glycerol can attach to 1, 2 or 3 fatty acids, and accordingly we will have monoglyceride, diglyceride and triglyceride, right? Now fatty acids can be esterified. The bond between the fatty acid and glycerol, that is ester bond. So we call it esterification. She told you monoglyceride, diglyceride and triglyceride. You know the difference? All of these, mono, di and tri, only one glycerol molecule. So the difference is based on the number of the fatty acids. Mono means one fatty acid attached, di is 2 and tri is 3. Are these points clear to all? Yes? Am I audible? Yes, yes, yes. Alright guys, now moving on here. So oils have the lower melting point, for example gingerly oil, and hence remains oil in the winters. There are some different types phospholipids. Remember we have yesterday we have studied about the cell membrane. The lipid which is most abundant in the cell membrane is phospholipid, found in the cell membrane. Lecithin is one example. So especially some tissues, especially the neural tissues, have lipids which are more complex structures. Now talking about the nitrogenous bases, we have 5: adenine, guanine, cytosine, uracil, thymine. Now if we add sugar to it, S for sugar, S, it becomes nucleoside, right? And if phosphate group is also attached or esterified to the sugar, it is the nucleotide, right? Now see the names of the nucleosides: adenosine, guanosine, thymidine, uridine, cytidine, nucleosides, and adenylic acid, thymidyllic acid, guanylic acid, this, and cytidyllic acid are nucleotides. Are these points clear? Now this page is quite important. See the structure of the sugars here, the amino acids, although I will come back to this, but the page is really important. Now coming on to primary and secondary metabolites, very important. So is this table. Pigments you should know. Which are the pigments? Alkaloids are morphine and codeine. Terpenoids are monoterpenes and diterpenes. Essential oils are lemon oil. Toxins, aberrin and ricin. Lectins, concanvaline A. Drugs are vinblastin and vincristin also, and curcumin. This is from haldi, turmeric. And polymeric substances come from rubber, gum and cellulose. One of the most important tables for your NEET exam, right? Now, what is the difference between the primary and secondary? You cannot survive without primary metabolites. They are essential for your physiological activities. While if we talk about the secondary metabolites, see, it is written primary has role in normal physiological process, while the secondary metabolites are not important for the host organism. They are useful to the human welfare, like rubber, drugs, spices, scents. All these are secondary metabolites, or some of them have ecological importance. Is it clear? Now coming on to biomacromolecules. Just see the structure. See, the compounds found in the acid soluble pool, I told you, they are micromolecules. What is the size of the micromolecules? Between 18 to 800 Daltons only, right? While the acid insoluble pool has macromolecules. It consists of the polymers, generally proteins, nucleic acids, polysaccharides and lipids. Lipids are those smaller in size but still found among the acid insoluble pool. Now see, with the exception of lipids, all of them, see, proteins, nucleic acids, polysaccharides, they have this molecular weight ranging in 10,000 Daltons or above. Is that clear? Now see, that's already told. Acid insoluble has macromolecules, right? Now why do lipids with molecular weight which does not exceed 800? So here it is clear, lipid is a micromolecule when it comes to the molecular weight, but it comes under acid insoluble pool. Why? So here is the reason given. So when you are crushing the cells, the membranes are broken into pieces. Therefore, these membrane fragments are formed in the form of vesicles, and those vesicles have a larger size as compared to the individual lipid size. Is it clear? You're not getting the individual fatty acid, you're getting them in the form of vesicles. So vesicles will have a larger size, separated along the acid insoluble pool. Is it clear? Now very important line, acid soluble pool represents roughly the cytoplasmic composition. So what does your cytoplasm contain? Acid soluble pool. Are these points clear? Another very important table which is telling you the average composition of different components in the cell. Now see, so water is maximum 70 to 90%. You have to remember the percentage as well. Proteins 10 to 15, carbohydrates 3, lipids 2, nucleic acid 5 to 7 and ions is 1. So there is a mnemonic. See, we can easily remember water is maximum. What is the mnemonic for rest? So it is P na ka li. Can you remember that? P na ka li. So this is the decreasing order. This is the decreasing order in which the components are present. So after water, so water is of course maximum. After water it is proteins, then it is nucleic acids, then it is carbohydrates, then lipids, and then finally we have ions. Is that clear? So remember this mnemonic. Maximum water, we remember pinakali. So pinakali will help you remember the decreasing order of these components. Is it clear? Now moving on to proteins here. So proteins we know is a polymer of amino acids, and how many different amino acids will participate? It is 20 amino acids which help in protein synthesis, right? Now because there are different amino acids combining, hence protein is not a homopolymer, it is a heteropolymer. It is a heteropolymer. Is that clear? Now these can be essential or non-essential. So those which you have to take in your diet, the dietary proteins are always essential. Which the body can produce on its own, that will be known as non-essential. Is that clear? Now see the functions. This table is another very important table in your NCERT. Very important. See the functions. You usually get question from this table, one or the other. Collagen, intercellular ground substance. Trypsin is a protein digesting enzyme. Insulin is a hormone, we know that. And then antibodies, they fight against the infections, receptors, sensory reception of all these stimuli. And GLUT4. GLUT4 stands for glucose transport 4. There are different types of GLUTs, right? Glucose transport proteins. So GLUT4 will enable the glucose transport into the cell. Is it clear? So you usually get question from this table, so make sure that you learn that very well. Now moving on to the next. Collagen is the most abundant protein in the animal world, right? While if we talk about the overall biosphere, it is rubisco that takes over. Rubisco, ribulose, bisphosphate, carboxylase, oxygenase, an most abundant protein in the whole biosphere. Now coming on to polysaccharides. See, we know that polysaccharides are polymers of simple carbohydrates, simple sugars. Now again, these can be homopolysaccharides also or hetero also. So only one type of sugar can just go on combining to form a bigger sugar, right? For example, see here, cellulose is a polymeric polysaccharide made up of only one type of monosaccharide. Not only this, so it is a homopolymer. So is starch and also glycogen. All glycogen, starch and cellulose, they are made up of one type of monosaccharide, which is glucose. They are all polymers of glucose. While inulin is a polymer of fructose. And remember, inulin is used for kidney function test. Is that clear? Because inulin is not metabolized by your body. Now, very important line here. In a polysaccharide, be it any, but they are given an example of glycogen here. In any polysaccharide, the right hand side is the reducing end. Can you remember RR? So there are many RRs in our bio, I have told you, right? Do you remember the previous session? We have discussed R for radical, R gives rise to root. Radial arrangement of vascular bundles in root, right? Then RRR, carrageen and red algae. Remember that? And then RR, right end will of polysaccharide is reducing in nature. So we have many RRs in our biology. Remember all these combinations. And the left end would be non-reducing. Is that clear? Now see here. Now we know that starch gives iodine test. We use iodine test for testing the presence of starch. So original color of iodine is brownish yellow, but in the presence of starch it becomes bluish black, right? Why? Starch forms helical secondary structure with iodine and holds the iodine molecule. Is that clear? Now starch iodine is blue. This complex between iodine and starch, that is bluish black in color. Cellulose will not give you any iodine test because cellulose does not contain complex helix and cannot hold iodine molecule. Is that clear? So iodine test is given by starch as well as glycogen. Although the coloration is different here, but cellulose will not give you that test. Is that clear? Now see the structure. This is the structure of glycogen. This is the structure of glycogen. Is that clear? Now, moving on here. So I already told you cellulose is also the monomer, also the polymer of glucose. Now see here, there are other sugars, modified sugars like glucosamine, N-acetyl galactosamine, etc. And there is this compound called chitin. Now chitin is a homopolysaccharide but of a different sugar, NAG. N-acetyl glucosamine. No, and NAG, that will form the chitin. And where do you find chitin? In the exoskeleton of arthropods as well as in the cell wall of fungi. Are these clear? Now all these are homopolysaccharides, I've told you. Now coming on to the nucleic acid, we already know there are two types of nucleic acids, DNA and RNA. The main differences of DNA and RNA will be discussed in the chapter of genetics. Although, let's see here. So we know that nucleotides, see, the building block is a nucleotide for any, be it DNA or RNA. Who's the nucleotide? It is, sorry, who's the monomer? It is a nucleotide, and nucleotide has three components: heterocyclic compound, that is the nitrogenous base; monosaccharide, a sugar, pentose sugar; and third is the phosphoric acid or a phosphate. Is that clear? Rest we have already discussed. The sugar is different. In DNA we have deoxyribose, and in RNA we have ribose. Is that clear? So guys, I have discussed all these details in my marathon. In case there is any doubt regarding any chapter, please quickly go and watch that at a faster speed. Is that clear? We still have time. Is that clear? And now, since I have completed the marathon, so syllabus is once done, and today we'll be completing class 11 and then class 12 also. It is high time that now you start taking these tests. You start taking these all India mock tests, and so we are getting nothing out of it. They are absolutely for your practice. So make sure that you do these. And also, if you want to do, take more practice at your own convenience, because honestly speaking, more than reading the things again and again, test taking, analysis will do more good at this point of time. If you are skipping a test even on a single day basis, that is not a good sign, to be honest. Is it clear? So make sure that you don't miss any test. All right, coming on to the structure of proteins, guys. So see, this proteins will have primary, secondary, tertiary, and quaternary structure. What is primary structure? It's like an imaginary linear line. It is a linear structure where a line represents left side is the first amino acids and the right end represents the last amino acid. So these lines are important. See, so the first amino acid will have free amino group, hence it is the N terminal, and last one will have free carboxyl group, hence it is known as the C terminal. See, in proteins, I'm talking about the secondary structure. When we say there is a alpha helix, so it is always the right-handed helix that is present. Is it clear? That is the secondary structure. Woolen ball-like structure represents the tertiary structure. It is the three-dimensional structure and very important for the biological activity. So all the enzymes will have tertiary structure, by the way. Quaternary structure, as observed in adult human hemoglobin, quaternary structure is when a protein is made up of more than one polypeptide chain. So when different polypeptide chains club together, so they give rise to a quaternary structure overall, right? Now in adult hemoglobin, we know there are 2 alpha and 2 beta chains, right? Please tell me, guys, that these points are clear to all. Right? Now moving on to the very important part, that is enzymes. All enzymes are proteins. Is it a true statement? No, because I have not used the term almost, because there are exceptions. So all enzymes are proteins? No, but almost all enzymes are proteins? Yes, right? Almost all enzymes are proteins, because some nucleic acids that behave like enzymes, for example, ribozyme, it is an RNA molecule, right? Now, see here. So there are some enzymes, see, mostly enzymes, they work maximum, their optimum temperature is between 35 to 40 degrees Celsius, but then there are enzymes which can withstand high temperatures, which are extracted from these hot vents and sulfur springs. They are stable and retain catalytic power even at temperatures up to 90 degrees Celsius. This thermal stability is a quality of such enzymes isolated from thermophilic bacteria only, right? Our enzymes don't have that property. Now moving on to the next, coming on to this part, let's talk about this paragraph. See, concept of activation energy, very important paragraph. First of all, you should take notice of what is taken along the y-axis. It is the potential energy, and what has been taken along the x-axis, this is the progress of reaction, right? Now in the potential, so there are 2 graphs drawn here. One graph is activation energy without enzyme, the other is in the presence of enzyme. Now see the activation energy without enzyme, it is of course higher, and in the presence of enzyme it is lower. So what is the function of enzymes? They lower the activation energy. So whether the enzyme is feasible, whether the reaction is feasible or not, it depends largely upon the availability of the activation energy, right? So enzymes do what? Enzymes lower that need for activation energy, and that is how they increase the rate of the reaction, they speed up the reaction, they catalyze it. Now the difference between S and P, S is the energy of potential energy of substrate, and P is the potential energy of products. So see, the energy difference between S, potential energy of substrate, and that of product will tell you whether the reaction is exothermic or it is endothermic. For example, now see, the energy of substrate is higher, that of products is lower. It means maximum energy has been released. So in this case, when energy of substrate is more, then it is always an exothermic reaction. If it is the reverse case, that means the energy of product is here and substrate is here. So products have higher energy, that means they have absorbed energy. Hence that would be an endothermic reaction. Is that clear? So exothermic, also known as spontaneous. And the difference between the average energy content of substrate and that of the transition state is known as the activation energy. I hope you know. This is the transition state. This is the energy of substrate. Difference between energy of transition state and substrate, this is called activation energy. So be thorough with all these statements. Now very, very important, you have got question from this topic multiple times. Which of the following is the correct sequence of enzymatic action? So first of all, see the reaction. Enzyme plus substrate will form enzyme substrate complex, will change into enzyme product complex, and product will separate from the enzyme. And this explains why enzyme is required in the very small amount, because enzyme will be re-available for next use, next lot. So first, what happens? Substrate binds to the active site of enzyme. Now, binding of the substrate induces the enzyme to alter its shape and fit more tightly around the substrate. You have to learn these in a proper sequence because it is a process, right? Next, active site of enzyme, now in close proximity to the substrate, breaks the chemical bonds. This is the third step. And the next in the sequence is, once the enzyme substrate are formed, they are tightly attached, what will happen? And substrate gets converted into product. Enzyme will release the product of the reaction, and the free enzyme is ready to bind to the another molecule of the substrate and run through the catalytic cycle once again. So these four steps have to be learned in sequence. NTA loves sequence based questions. Let's move on. Now coming on to the factors affecting the enzymatic activity. So temperature, pH, concentration, and then we'll also talk about some molecules which can inhibit the reaction. We call them inhibitors. First, let's see temperature and pH. See, temperature preserves, low temperature preserves the, so underline this also, low temperature preserves the enzyme in a temporarily inactive state, and high temperature will destroy it because proteins are denatured by heat. What does this indicate? That neither the enzymatic action will occur on very low temperature nor it will occur on very high temperature. So we need a small range within which it can perform to its maximum, its best. That temperature is called the optimum temperature. Is that clear? You decrease the temperature below the optimum, again the enzymatic activity will decline, and you increase beyond the optimum, again it will decline. But the decline beyond the optimum temperature will be permanent. That is called denaturation. Now the same trend is also seen in case of pH. So there is a fixed pH at which the enzyme will perform to its best. That is optimum pH. Every enzyme will have different type of optimum pH. Is that clear? So what is the type of graph that you get? Be it temperature, be it pH. You take enzymatic activity along the y-axis and pH or temperature along the x-axis. See what is happening here. So that this is the maximum activity. This is the maximum activity, right? So this pH is known as optimum pH. This temperature is known as the optimum temperature. Do you observe that beyond this, below and beyond the optimum pH or temperature, the activity is declining. And as far as temperature is concerned, we use the Q10 law. What does it say? With every 10 degree increase, the enzymatic activity doubles. That happens only till we reach the optimum temperature. Are we clear with these things? We have discussed it all in marathons, guys. So Next, coming on to concentration of substrate. Again, see the graph. Along the y-axis, you should be very clear about these graphs, guys, that what is being taken along the y-axis and what along the x-axis. Now see here, along the y-axis, we are taking the velocity of the reaction. Along the x-axis, I'm taking the substrate concentration. S in square parenthesis. Parenthesis means brackets. When there is a square bracket around the S or P, it indicates their concentration. Here we are taking the concentration of substrate molecule. Is that clear? Now, so do you see, initially as I am increasing the concentration, velocity is also increasing. But this happens only to a certain amount, only up to a particular substrate concentration, beyond which you see that the line goes parallel to the substrate. That means you increase the substrate concentration, but beyond a certain point, velocity of reaction will not increase. Why? Why? It is clearly written here. See, the velocity of the enzymatic reaction rises at first, reaches a maximum velocity and not exceeded by any further increase in concentration, because enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no enzyme molecules to bind with the additional substrate. So all the enzyme molecules have already exhausted. They are all at work. So till the product is not formed and it is not released, now more substrate concentration will not help in increasing the velocity, will not cause increase in the velocity. Is that clear? So this is the Vmax, and correspondingly we will get Vmax, half of Vmax here. And very important, the substrate concentration at which the half of Vmax is obtained, this substrate concentration is known as Km, or Michaelis-Menten constant. Is that clear? Now, fourth factor is the presence of certain molecules which can just pause or completely end the reaction. Those molecules are known as inhibitors. They will inhibit. So there are different types of inhibitors, though here it is only one type of inhibition that is mentioned, that is competitive inhibition. So inhibitor which closely resembles a substrate in its molecule. See, competition is when two things are fighting for the same thing. That is when competition arises, right? Now, the inhibitor molecule and substrate molecule, they both look similar. They are geometrically similar. They have similar configuration, right? Because of the similarity in their structure, so instead of substrate binding to the active site, it is the inhibitor that binds and it inhibits the activity of the enzyme. This is competitive inhibitor. Very important example here is that inhibition of succinic dehydrogenase by malonate. Now this is the reaction of Krebs cycle actually, right? Instead of, see, succinic dehydrogenase has to bind with succinic acid. But instead of succinic acid binding to this dehydrogenase, it is malonate. And when malonate binds, it will stop the reaction. Is that clear? And another example, this mechanism is also employed to stop the bacterial growth. So we use competitive inhibitors as often used in to control the bacterial pathogens. There are some sulfa drugs which are working in this pattern. Is that clear? Now talking about the classification, see, enzymes are divided in 6 classes, and how do we remember? We remember it as OTH-LIL. OTH-LIL. You have to remember it in sequence. You have to remember it in sequence. What is the sequence? OTH-LIL. Right? Oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Clear? Now, oxidoreductases, as you can see, it will be involved in reduction oxidation reaction. Transferases will help in transfer of group. See, it was SG and S dash. Now this G has been transferred to S dash. Hydrolases, hydrolytic. So in the presence of water, they are going to break down and cause hydrolysis of ester or ether or peptide or glycosidic and so many bonds. Is that clear? But they require water for doing that. While there are lyases, they are also involved in breakdown, but without involvement of water, and instead they will cause double bond formation. See the reaction here. Is that clear? So this reaction should be well identified by you, well observed. So you should know what is the difference between the two. Now coming on to the fifth type, that is isomerases. Of course, they will help in isomerism. be it of any type, and then ligases. It will help in the linking together of two compounds, be it binding of the two DNA fragments, or CO group, or CS, or CN and PO bonds. Is that clear? So how do you remember this? Now, very important line, the enzymes are divided into 6 classes. So this is class 1, class 2, class 3, 4, 5 and 6. So actually enzymes have been given a number, a 4-digit number or so. So, right? So these enzymes are given this 4-digit number. The first digit, let's, for example, I say 4321. So this number indicates that whatever the enzyme be, it 100%, it definitely belongs to this class of liases, 4th class. So this is how you use this number to identify the class it belongs to. Now, second number indicates the subclass. It indicates the subclass. Now, each enzyme or each class is further subdivided into 4 to 13 subclasses. We don't have to bother to learn that, but just remember it. Every enzyme will have a 4-digit number. First number indicates the class and the second number indicates the subclass. Is that clear? Now, coming on to yes, coming on to the last part, which is cofactors. See, there are certain enzymes which are not just made up of proteins, right? Which have only protein component, are simple ones. But then there are other enzymes which require a non-protein component, and that is called a cofactor. And without the binding of this cofactor or the non-protein component, it will not provide catalytic activity. Now see here, the cofactors are of 3 different types. Okay, so the protein part is known as apoenzyme and the non-protein part is known as cofactor. Now cofactors are of 3 different types: prosthetic group, coenzyme and the metal ions. Prostatic groups are organic in nature, tightly bound to the apoenzyme. Very important. Peroxidase and catalase. Very important example. Already been a PYQ. So peroxidase and catalase, what do? They catalyze the breakdown of hydrogen peroxide to water and oxygen. Right? And what is the cofactor required for the activity of both these? It is heme. So until and unless heme binds to peroxidase and catalase, it will not catalyze the breakdown of hydrogen peroxide. Secondly, we have coenzyme. They are organic, but they are transiently bounded, right? So it will only bond very temporarily with their apoenzyme. For example, see, many coenzymes are vitamins. Why do we need to eat vitamins? Because they have these regulatory functions, right? So coenzyme NAD or NADP contain the vitamin niacin. Is that clear? Now, number of metal ions are also working as cofactors. For example, they form coordination bonds with the side chains of the active site. For example, zinc is important cofactor for the enzyme carboxypeptidase. This is a protein digesting enzyme. So what does it indicate? If you will not eat zinc, you can't digest your proteins properly, right? Now, catalytic activity is lost when the cofactor is removed. Please let me know that this chapter is well read and there is no doubt about it. Yes, guys. And with this, we start the next unit, which is plant physiology. Yes, guys. Quickly let me know. Are we clear? Can we start with chapter 11, which is photosynthesis? Yes or no? No doubt? Cool? Yes, guys. Quickly let me know if there is any doubt. All the serious students, give me a thumbs up that we are all clear, all read. All right. See, today there is no upper limit because Wasim sir has class at 8 p.m. today, right? So we are going to complete the chapters somehow. All clear? Ma'am, cell cycle division. Heota, we have done that yesterday. Please watch the last part of the marathon. We have done that yesterday already. Yes? All right, guys. Let's move on to plant physiology, starting with the first chapter, one of my favorite chapters, photosynthesis. Let's start here. So, so we are starting with the experiment. So variegated leaf experiment. Variegated leaf means a leaf which is multicolored, right? When it is exposed to light, what happens? Photosynthesis, it indicates that photosynthesis occurred only in the green parts of the leaves in the presence of light. So this is the experiment which we have already conducted in class 9th or 10th. So we are just revising it here. So variegated leaf means multicolored, and multicolored leaf will indicate that photosynthesis will occur only in the green parts. What can we conclude out of it? That chlorophyll is important for photosynthesis, right? Basic experiment. Now, next experiment again done in class 9th or 10th, that is leaf when enclosed in a test tube containing KOH. What will happen? See, KOH, potassium hydroxide, is going to absorb CO2, and this experiment will explain that CO2 is required for photosynthesis. The leaf which has been exposed to KOH is devoid of carbon dioxide, right? KOH is absorbing all the carbon dioxide. So leaf is not getting CO2 to perform photosynthesis. It will not give that iodine test. It will not produce starch, right? So what does this indicate? KOH test, that CO2 is required for photosynthesis. Now coming on to these experiments done by Joseph Priestley. He's the one who discovered oxygen, right? Discovered oxygen, did this bell jar experiment, and what does he conclude? That plants restore the air whatever breathing animals and burning candles remove, right? Because we see here that when the plant was mint plant, and which plant did he use? He placed the mint plant here, okay? When the mint plant was not placed in the jars, both of them died. The candle also got extinguished and the mouse also died after a while, but mint plant kept both of them alive. So see, that is why he concluded that plants restore to the air whatever the breathing animals and burning candles remove. Is that clear? Now, using a similar setup as that used by Priestley, it was Jan Ingenhousz who showed that sunlight is essential for the plant. Is that clear? And it was Ingenhousz who discovered this, or sorry, who used this term photosynthesis. Is that clear? Now, Ingenhousz also did another experiment with aquatic plants, showing that in bright sunlight, small bubbles were formed around the green plants, while in dark, the same setup did not show the presence of bubbles. Why? Later on, it was observed that these bubbles are nothing but oxygen molecules, right? So oxygen will be produced only so he showed that it is only the green parts of the plant that could release oxygen. Is that clear? Now, it was Julius von Sachs provided in evidence for production of glucose when plants grow. Second, glucose usually stored in the form of starch. And third, he also showed that chlorophyll is located in the special bodies known as chloroplasts. So Sachs has told us three things. Glucose is produced as a result of photosynthesis, it is stored in the form of starch, and this chlorophyll is present in chloroplast, right? Then talking about Engelmann. Engelmann did this prism experiment. So he illuminated the green algae. Illuminated means he showed light to green alga cladophora. Now cladophora is it is a green alga, right? So cladophora, when it was illuminated, of course it photosynthesized, and as a result of photosynthesis, oxygen would be produced. And when he placed cladophora in a suspension of aerobic bacteria, so bacteria multiplied, it grew quickly, but only that part of this aerobic bacteria grew which was present in the zone of only blue and red light. So bacteria eliminated mainly in the region of blue and red light. So he gave the first action spectrum. Spectrum of chlorophyll A and B have been discussed later. Please tell me these points are clear to you all. Is that clear? Now coming on to next part, that is Cornelius van Niel. Van Niel, based on his studies of purple and green bacteria, photosynthesis is essentially a light-dependent reaction, and hydrogen from a suitable oxidizable compound reduces the CO2 to carbohydrates. Please tell me these points are clear to everyone. Yes, guys, please tell me these points are clear to everyone. Right? Now, so in green plants, the present plants, water is the hydrogen donor and oxidized to oxygen, while in some organisms H2S is the hydrogen donor, for example in purple and green sulfur bacteria. So instead of oxygen they produce sulfur as a byproduct. Is it clear? So oxygen evolved by the green plant comes from water and not from CO2. This was later proved by some radioisotopic techniques. Are these points clear? Now coming here, where does it take place? So you must remember, in stroma, enzymatic reactions are going to synthesize the sugar. So there are two reactions. One is light reaction, also known as the photochemical reaction. Where does this occur? It occurs in the thylakoid or the grana or the stromal lamella. While dark reactions, these are the carbon reactions, assimilatory reactions basically. So biochemical reactions also we call them. So these reactions occur in the stroma. Is that clear? So this is the structure of chloroplast. So you should well observe it, and we'll be discussing the diagrams separately. Now see, these graphs are very important, very important. So this is the absorption spectrum. See, three pigments have been discussed here. So one is chlorophyll A, the first peak, right? This is chlorophyll A, the second peak of chlorophyll B, and these multiple peaks of carotenoids. So you should be able to label them first of all. Also observe that in which region, so on the y-axis guys, on the x-axis we are taking the wavelength of the light in nanometers. So if we see here, so it is in the red and the blue light that a spike is seen in the absorption spectrum. Is it clear? So it shows that the maximum light absorbed by the pigments is either red or it is blue. Now, if we perform paper chromatography, we will see different types of pigments present in the plants. For example, chlorophyll will show, very important, this is a PYQ. You got this question in match the following. Match the following. Chlorophyll A, bright or blue-green. Chlorophyll B, yellow-green. Xanthophylls, yellow. Carotenoids, yellow to yellow-orange. Very important. So can you now tell which is the most abundant pigment in the world? It is chlorophyll A. We already know that. So while this is absorption spectrum, guys, this is the action spectrum because it shows the rate of photosynthesis. So now if we observe carefully, so the rate of absorption and the rate of actions, so absorption spectrum and action spectrum, they are overlapping, and this overlap has been indicated in this third graph here. So wherever we saw the maximum absorption in the same region, we are able to observe the maximum action as well. So the more the light absorbed, more is the photosynthesis, and it happens in the region of red and blue light. Is that clear? So color of light at which, even now when we have to compare between red and blue, the red takes the lead, right? So maximum absorption occurs at the red light. So is the maximum absorption A in the blue or the red? The answer is red. Out of the two, it is red again. Now see here. So talking about the light reaction. Light reaction, also known as photochemical reaction. What are the processes that are going to occur in the light reaction? Number one, light absorption. Second, water splitting. Third, oxygen release. And fourth, formation of high energy chemical intermediates. In high energy chemical intermediates are ATP and NADPH. So these are the four things which occur during light reaction. So can you get a question like this? Which of the following is not occurring during light reaction? Or which of the following is not the event during light reaction? So you should remember these four steps. What are these? These are light absorption, water splitting, oxygen release, and formation of high energy intermediates. Is that clear? These are the four important points or the things that occur during light reaction. Now for light to be absorbed, guys, we need some pigments organized in a proper system. So we call this light harvesting complexes present within photosystem 1 and photosystem 2. So light harvesting complexes are made up of 100 of pigment molecules bound to the proteins, right? And harvesting complexes systems are also known as antenna molecules. So there is a single chlorophyll A molecule which is known as the reaction center, and around the reaction center it is the other accessory pigments and proteins. Reaction center is different in both the photosystems. In PS1, photosystem 1, reaction center of chlorophyll A has maximum absorption peak at 700 nanometer. So we call it P700. While in PS2, the absorption maxima occurs at 680 nanometer. Is that clear? So PS1 700, PS2 it is P680. Are we clear? Now talking about the electron transport. See, what does these reaction centers consist of? Single molecule of chlorophyll A. Clear? Now see, very important, very important. So the movement of electrons is downhill in terms of oxidation, reduction or redox potential. So if the electron is rising from this chlorophyll A molecule, so movement is going to be downhill in terms of oxidation, reduction, reaction or redox potential scale. Simultaneously, electrons from both PS1, PS2 and PS1 will be released simultaneously. Is that clear? Now see here, the whole scheme of transfer of electrons starting from PS2, uphill the acceptor, down the electron transport chain to PS1, and again excitation of electrons and transfer to another acceptor, that is FD, and finally downhill to NADP. So electron will be given to the next one with low potential, downhill to NADP reducing. Is this clear? Then, so because there is uphill, downhill and uphill, this movement is known as Z scheme because of its characteristic shape. Are we clear? So this is the Z scheme of electron flow, and in Z scheme of electron flow, what do you see? Both PS2 and PS1 are involved. Is that clear? Also guys, it will involve the function of NADP, and NADPH will be produced as well. Is that clear? Now splitting of water. How does PS2 supply electrons continuously? So we know when light is falling on PS1 and PS2, electrons are continuously getting released. So if the electrons are only getting released and not compensated, then the chlorophyll A will cease to exist, right? It will no more be present. After all, it is all made up of electrons, protons and neutrons. So electrons are all lost. Chlorophyll ceases. Now, so we need to continuously supply that loss of electrons. So who is going to be the supplier? It is the water. Water splitting complex is associated with PS2. Now these are the questions. So water splitting complex is associated with PS2. So where is it present? In the lumen? No. It is present on the inner side of thylakoid membrane, very near to this. Is that clear? So we have seen all those things. See, it is associated with PS2, which it is physically located on the inner side of the membrane, not in the lumen. So all these points are very, very important. So now we have cyclic and non-cyclic photophosphorylation. It is ATP production in the presence of light. Now, if the electron flow is Z scheme, it is non-cyclic. If it is cyclic, then it will be cyclic photophosphorylation. See here. So when the two systems work in series, first PS2 is used and then PS1, process is known as non-cyclic because the electron once lost is not coming back to the source, right? It is called non-cyclic photophosphorylation, occurs during the Z scheme. While when only PS1 is functional, the electron is circulated within the photosystem, comes back to the source, right? This is called cyclic flow of electrons and occurs in the stromal lamella, while this Z scheme occurs in the thylakoid membrane. Is that clear? So the membrane or the lamella of the grana have both PS1 and PS2, and PS2 of the stromal lamella, while the grana has both PS1 and PS2. The stromal lamella membranes lack PS2 and as well as NADP reductase. So neither neither NADP formation takes place here, nor the water splitting and oxygen release occurs during this stromal lamella membrane. Is that clear? Now the cyclic flow results only in the synthesis of ATP, not of NADPH. Is that clear? And only cyclic photophosphorylation absorbs only light of wavelengths beyond 680 nanometer, because the maximum spectrum is that of 700, right? So beyond 680 nanometer are available, only then it will function. Very important lines. Now talking about chemi-osmotic hypothesis has been put forward to explain the mechanism of ATP synthesis. Very, very important. See here, this is a PYQ. In 3-4 years, we have got this question continuously. See, what does chemi-osmotic hypothesis explain? It is related to ATP synthesis. Now, what is required for ATP synthesis? We'll understand. So, first of all, ATP synthesis is linked to the development of proton gradient across the membrane, across the thylakoid membrane we are talking about. Now, since splitting of water molecule takes place only on the inner side of the membrane, that is within the lumen, hydrogen ions are produced by splitting, accumulate within the lumen. So within the lumen, we have high concentration of protons. Is that clear? See here. So this is where water splitting occurs, that is towards the lumen. Towards the lumen. I did not say inside the lumen. It occurs on the inner side. But all these H positive ions produced, they will be accumulated in the lumen, yes or no? And on the stromal side, we will have relatively less amount of H positive ions. Yes? Now see. Primary acceptor. Another reason why the level of protons increase within the lumen. See here, we have discussed that the primary acceptor of electron, which is located towards the outer side of the membrane, transfers electron not to the electron carrier but to the hydrogen carrier. What is the hydrogen carrier being talked about? It is the plastoquinone. Right? So pheophytin, that is the first primary acceptor of PS2. Pheophytin doesn't give this electron to any electron carrier. It passes it on to plastoquinone, PQ. Now, plastoquinone is not meant for electron transport. So, if you want PQ, that is plastoquinone, to do that for you, you have to give some favor to it, right? So, what does how does this hydrogen carrier transport the electron? So, see here, this diagram. So when the electron reaches here, pheophytin is going to give it to PQ, plastoquinone. Now in return, plastoquinone wants that you should allow it to pump one hydrogen proton from the stroma into the lumen, right? And in return of that, it will give you that favor of transporting the electron to cytochrome B6F. Yes? So, plastoquinone is going to pump hydrogens into the lumen. So, two reasons why the level of protons are increasing within the lumen. One is because of water splitting and second is because of plastoquinone pumping it in return of electron transport, because itself it is a hydrogen carrier. Is that clear? And also, this level of H positive ions, protons, are going to reduce in the stroma because of NADP, because it is continuously consuming H positive ions to make NADP, H plus H positive. Is that clear? So, is that clear? So, protons in the stroma decrease in number while in lumen it is accumulating. Now, breakdown of this gradient leads to synthesis. Very important. So, when you break down this gradient means difference. It is during the breakdown of the gradient that ATP will be produced. See here, due to the movement of protons from the membrane, across the membrane, sir, that means from the lumen to the stroma, through the transmembrane channel of CF0 of ATP synthase. And the ATP synthase consists of two parts, CF0 embedded in the thylakoid membrane forming a transmembrane channel carrying out the facilitated diffusion, and the other portion is CF1 which is protruding out towards the stromal side. Is that clear? So chemiosmosis requires, this is your PYQ. This is your PYQ. So chemiosmosis requires a membrane. Which of the following components are required for chemiosmosis? That was the question. So we need a membrane, we need a proton pump, we need a proton gradient, and we need ATP synthase. These are the four components required. So energy is used to pump out protons from the membrane to create the gradient, and then high concentration of protons within the thylakoid membrane. ATP will be used immediately in the biosynthetic process, that is in the dark reaction, which will be responsible for CO2 fixing and synthesis of sugar. Now, where are these ATP and NADPH used? So, they will be used in the dark reaction. See here. Now, use of radioactive C14 by him. We are talking about Melvin Kelvin. In the algal photosynthesis studies led to the discovery that the first CO2 fixation product is the C3 carbon compound. Now, it was Melvin Kelvin who gave us that process, how does dark reaction occur. And he concluded that the first stable compound produced during the reaction is a 3-carbon compound. Hence we call dark reaction or Kelvin cycle as C3 cycle also. Is that clear? So the first compound was identified as 3-PGA, 3-phosphoglyceric acid, PGA, 3-PGA. How many carbon atoms does it have? 3. So all the cycles that we are going to study, C3, C2 or C4, they are based on the number of carbon atoms present in their first stable product. Is that clear? Now, so PGA as a first product of CO2 fixation. The first stable product of CO2 was again an organic acid, but in case of C4 plants, guys, it is going to be a 4-carbon compound. I just told you. The name of the cycle is based on the first stable compound, right? If it has 3-carbon compound, we'll call it C3. If it'll have 4-carbon compound, like in case of C4 cycle, 4-carbon compound, oxaloacetic acid, we call that cycle as C4. And in case we'll get 2-carbon compound, it will be known as C2 cycle. We'll come on to that as well. Is that clear? So in C3 cycle, it is PGA, 3-carbon. In C4, it is OAA, oxaloacetic acid. You should remember the names of these first stable compounds very clearly. Now, the primary acceptors, see, of carbon dioxide. If we are talking about C3 cycle, guys, the answer is ribulose bisphosphate, RUBP, and this is itself a 5-carbon compound. This is in case of C3 cycle. C3 cycle. Is that clear? And in case of C4 cycle, guys, in C4 cycle, it is going to be PEP. And PEP is itself a 3-carbon compound. Is that clear? So CO2 will be absorbed by this 5-carbon compound and forming 2 molecules of 3-PGA. We'll come to that. So let's discuss more about Calvin cycle here. So Calvin cycle, light reaction, these will occur in all the plants. See, it is clearly written. It occurs in all the photosynthetic plants, right? It does not matter whether they are C3 or C4 plants. Even if it is a C4 plant, ultimately C3 cycle has to occur because the sugar formation, the food formation, the biosynthesis will actually occur during C3 only. Is that clear? Now, there are 3 steps in every C3 cycle known as CRR. Carboxylation, reduction and regeneration, right? So what happens in carboxylation? It is addition of CO2, and who's going to be the acceptor? If we talk about C3 plants, it is RUBP, utilized by carboxylation of RUBP and catalyzed by the enzyme Rubisco RUBP carboxylase oxygenase. Is that clear? They have not used the word oxygenase because here Rubisco is showing the carboxylase activity. So see, more correct to call it carboxylase oxygenase, Rubisco. Now moving on to the next step. So see, this is the cycle. This is the cycle. This is RUBP getting carboxylated, so adding CO2, and so reaction is known as carboxylation, and it is forming 3-PGA. So you must know that this is a 5-carbon compound, this is 1 carbon total, 6 carbon, but this is a 3-carbon compound. So in order to maintain and follow the law of conservation of mass, so there has to be 2 molecules of 3-PGA. Is that clear? The reaction is called carboxylation. You got the exact same cycle in your exam. This is a PYQ, and these highlights are the questions. So you have to put it in the correct sequence. The first question from the cycle is, which of the following is the correct sequence of events? So carboxylation, reduction and regeneration. Then also observe that where is ATP getting utilized during the C3 cycle. So 1 ATP is used in conversion of 1 molecule of 3-PGA into triose. Since we have 2, so 2 molecules of ATP are used during the reduction reaction. Yes, and 1 molecule of ATP will be utilized during the regeneration reaction. Also during reduction, we are utilizing 2 molecules of NADP. Is that clear? So 2 molecules of ATP, 2 molecules of NADPH utilized during reduction, and 1 molecule of ATP used during the regeneration. Simple. So in all, 3 molecules of ATP and 2 molecules of NADPH. every year, almost every year, we are getting numericals based on this question. Yes or no? Did we get this question this year? Let me know if you have gone through the paper in case. Yes. Yes, guys. Are we clear? Yes, Anurag. Why are you completely confused about it? If you are completely confused about this topic, Anurag, I am strongly recommending you to go and watch that marathon where we have discussed this topic in detail. Please go and watch that session. Is that clear? And come back and let me know whether it was clear to you or not. Yes, please. All right. So now during this 1C3 cycle, there was one triose phosphate molecule produced, right? Which will, now there are from 2 molecules of 3-PGA, we'll have 2 molecules of triose phosphate. So in total 6. We will take out 1 carbon. We'll take out 1 carbon, and in return, we'll have 5 carbons in for RUBP regeneration. Yes. So every C3 cycle is giving us 1 carbon, and in total, how many cycles should run? 6 cycles should run, where we will get C6H12O6. So for the formation of 1 molecule of glucose, 6 C3 cycles should run, right? So 6 C3 cycles means how much ATP will be utilized and how much NADPH will be utilized here. So it is clearly written. See, all these lines are important. So every CO2 molecule entering, 3 molecules of ATP, 2 NADPH are required, 1 molecule of glucose, 6 turns of cycle are required. And in the 6 cycles, 6 CO2 are entering to give us 1 molecule of glucose. In return, 18 ATP are used, 12 NADPH are used, and 18 ADP and 12 NADP are produced. Are these points clear? All these are PYQs, guys. All these are. Now, coming to C4 cycle, C4 is the oxaloacetic, is the first stable compound formed, and hence the name of the cycle is C4. Now, C4 plants are special. They have a special type of leaf anatomy, and they show high response to high light intensity, and they lack a process called photorespiration, and hence have a greater productivity. Now, this is where C4 plants stand out and have greater advantage as compared to C3 plants, because they don't perform C2 cycle. C2 cycle or photorespiration is one and the same thing. Photorespiration is a complete wasteful process. There is nothing good coming out of it, right? So rather it is just lowering the efficiency of Rubisco, and RUBP is getting wasted. So what is the special type of anatomy here? Leaf anatomy here. So vascular bundles. In C4 plants, they have bundle sheath cells, and we have read it in anatomy that bundle sheath cells are made up of sclerenchyma. So sclerenchyma has a thick wall. Now the wall is thick enough to not allow diffusion of any gas. Remember? Okay. So this arrangement where vascular bundle is surrounded by thick sclerenchymatous bundle sheath cells, that is called Krenz anatomy. And C4 plants, see the example, maize, sorghum. Basically, many monocots are C4 plants. I did not say all. For example, rice won't be a C4 plant. Is that clear? And few dicots are. But all the C4 plants, they are highly adapted for extreme temperature and high light intensity conditions. Now see, the primary CO2 acceptor is a 3-carbon molecule. I've told you already. It is PEP and present in the mesophyll cell. See, C3 cycle in case of C3 plants is occurring in the mesophyll cell. But in case of C4 plants, the C3 cycle occurs in the bundle sheath cell. And what happens in the mesophyll cell? Then mesophyll cell is going to run the C4 cycle. So first CO2 acceptor is PEP, and the enzyme used during the process is pepcase or pep carboxylase. See, mesophyll cells lack Rubisco, and OAA is formed in the mesophyll cell. Is that clear? So see the reaction here. All the reactions. So this is a mesophyll cell, this is a bundle sheath cells. In mesophyll cell, atmospheric carbon dioxide is getting added to PEP in the presence of pepcase, forming the first compound C4 acid. OAA. Now this OAA will change into another malic acid or aspartic acid and get transported into the bundle sheath cells, where again decarboxylation would occur to give rise to a C3 acid, pyruvic acid. Now this pyruvic acid will be transported back for the regeneration of PEP. Is that clear? So during decarboxylation, the CO2 being produced in this bundle sheath cell, it is making it a gas chamber, it is making it a CO2 chamber. So basically, why are we doing this? We are trying our best for Rubisco, yes, to show carboxylase activity and not oxygenase activity, because oxygenase activity is leading to photorespiration, a wasteful process. Is that clear? So this CO2 being produced, it is going to shift the priority of Rubisco towards CO2. More the CO2, more will be the carboxylase activity. Is that clear? Because Rubisco has a dual nature. See here, photorespiration. Important difference between C3 and C4 is photorespiration. Rubisco is the most abundant enzyme in the world. We have read that. In C3 plants, some O2 does bind to Rubisco and hence CO2 fixation is decreased. And RUBP, instead of converting to two molecules of PGA, binds to oxygen to form one molecule of phosphoglycerate and phosphoglycolate. Now see here, this phosphoglycolate is a 2-carbon compound, while this is a 3-carbon compound. And that is how the photorespiration gets this name C2 cycle, because apart from C3, there is a 2-carbon compound being produced here. So this helps us in differentiating. So which is a 2-carbon compound? It is phosphoglycolate, and that is photorespiration. In C4 plants, photorespiration doesn't occur. C4 plants lack photorespiration, hence productivity and yields are better, and they show tolerance to higher temperature. All these lines are very, very important. Is that clear? Now this table I have discussed in detail in that chapter, in my marathon. In case of any confusion, guys, please watch that marathon. Is that clear? We have discussed botany and zoology class 11 as well as class 12 in detail. Honestly, when I joined, like before your 3rd May exam, I didn't have enough time for revision, but now before Re-NEET, I have discussed everything in detail. Right? So you know that I joined the last days before your NEET exam. Can you please take a session on YouTube about all the questions you solved in the special classes? It is my sincere request. Yes, Jyoti, I can do that, but it is my humble request also. Why don't you join us in the special classes as well? Jyoti, please tell me first. Why don't you also join in the special classes? Let me complete few important sessions here, and I will bring few question sessions also on YouTube. But the point is, why don't you join us in regular special classes? Tell me, Jyoti. Okay, Kirta, I'll do that. Yes, there is a mock test today. There is 100% a mock test today, Hayota. You don't get mobile. Please request your parents, and see, actually you have to make your parents sit down and explain that the coming time we might shift on to, not might, there are huge chances that we'll shift to CBT, computer based test, right? Not only in NEET, even in higher exams also. So technology is not bad. So right now, if you're watching this session, you can take the test also. Request your parents. Sit in front of them and study so that they have more trust, right? That you're not wasting your time. Hindi classes running in Unacademy Learners app. No, Venkatesh, there are Hindi as well as English classes. Both the classes are running. So I am taking English classes only. Okay, I am taking English classes only. It's just that when there are few Hindi speaking students also attending, then I explain it in Hindi sometimes, but otherwise I'll take English classes only. Okay, all right. Now, the factors which affect the photosynthesis: availability of sunlight, temperature, CO2 and water. Blackman's law of limiting factors. See, if a chemical process is affected by more than one factor, then it is the rate which then the rate is determined by the factor which is nearest to the minimal value. That means the factor which is in minimum amount, that is going to be the rate determining factor. The least available factor is the rate determining factor. Now see this graph, very important graph. It shows the graph, it shows the light intensity affecting the rate of photosynthesis. See, initially we see a linear relationship between incident light and the carbon dioxide fixation rate. So this is only till the light intensity is low. So linear relationship till in low light intensity. But as soon as the light intensity goes beyond a certain level, that means it becomes higher, we do not see a rate increase in the rate of photosynthesis. Why? Because light saturation will occur at 10% of the full sunlight, right? So except for the plants in shade or dense, the shade loving plants, light is rarely a limiting, otherwise, so for shade loving plants, light is a limiting factor, otherwise light is usually not a limiting factor. It is plentiful. Is that clear? Now increase in light incident beyond a certain point, why it does not increase further? Rather, it will also show a decline. If we go for further study, we will see it not only becomes parallel, it will also show decline. Because see, because beyond a point, the breakdown of chlorophyll A will also occur and it will cause decrease in the photosynthesis. So after a while, actually, there will be this graph. The graph will decline like this. Is that clear? So this is what the last line indicates. Now talking about CO2 concentration, see, increase in CO2 concentration only till 0.05. Till 0.05 can cause increase in CO2 fixation. So you increase CO2 concentration, rate of photosynthesis increases, but only till concentration level of 0.05% in atmosphere, right? Beyond which it will also become toxic. C3 and C4 respond differently. See, at high light intensity, both will show increase. C4 plants show saturation at 360 value, while C3 plants will show saturation beyond 450. Why? Because in C4 plants, there is that Krenz anatomy. Remember, there is already a concentration, CO2 concentrating mechanism. But in C3 plants, they are already lacking it. Fine. So they will show saturation beyond 450. Is that clear? Now C3 plants respond to higher CO concentration and show higher productivity. And this has been utilized in some greenhouse crops like tomatoes and bell peppers. So what does that show? They are C3 plants, but by using this mechanism, by increasing the CO2 concentration, we are increasing their yield. And coming on to temperature. So remember, it is not the light reaction which is greatly influenced by increasing temperature. It is always a dark reaction, because temperature will affect the enzymatic activity. And enzymes are basically used during the C3 cycle or the C4 cycle, that means the dark reaction. So C4 plants respond to higher, and C3 plants have much lower temperature optimum. Is that clear? So tropical plants have higher temperature optimum as compared to the temperate climates. Water is one of the reactants, doesn't directly affect the rate of photosynthesis, but water stress can break down the stomata. Sorry, it can cause the stomata to close. If stomata will close, there won't be gaseous exchange. So there will be deficiency of CO2 also, right? So water stress doesn't directly impact photosynthesis, but indirectly it does. So for example, water stress will make the leaves wilt, surface area will decline, right? And for metabolic activities will also decrease. So guys, this was photosynthesis. Please let me know that this chapter is well read by you. And next, we have the chapter of respiration in plants. Quickly give me a thumbs up that we are all set for this next chapter. Yes, guys, quickly. Are we all ready? Quick. Give me a thumbs up. We are all ready for this chapter, all the serious students. If I start now, how to study, Shalini? First of all, tell me your level of preparation. Where are you right now? What were you doing before this? Why haven't you studied before this? I need to understand so many things before I answer this question. Honestly, guys, see, we do make such videos like how to improve your marks, even if you're scoring 250 right now, how to reach to 340 or 350, 40 plus, right? We do make such videos and we honestly do that. But to be very honest, these videos are only for, these strategy sessions are only for those students who have been giving their honest attempt, right? Them who have been making an honest effort. You have been studying, but somehow you didn't know where to improve. So you were not able to analyze things properly, and that's where we come in. We help you analyze your shortcomings. But if you are asking me that you have not studied at all and how to crack NEET in last 15 days, then anyone who is telling you such strategies is making a fool out of you. Do you think NEET can be clarified and cracked in just 15 days? So there is a topper who's been working so hard for 2 years and then 15 days you will crack NEET. How is that possible? Isn't that so? So please don't ask for such strategies. But yes, if you have been studying well, if you have been trying, and still your score is not improving, then I can help you. I can still help you. Okay? But you were sleeping till now, or whatever the situation was. Maybe you have some genuine situation. But then, whatever the reason be, if you have not studied anything till now, then last 15 days won't help. What you can do right now is aim for 2027, but don't waste these days as well. That's my suggestion. Okay? All right, guys. Coming on to cellular respiration here. Okay. Coming on to cellular respiration. What is water stress? Water stress means water deficiency. Water deficiency is known as water stress. Okay. So usually when we have deficiency of something, it leads to stress, right? So water stress is the same thing. Deficiency of water. Clear? Okay. Now talking about cellular respiration. What is cellular respiration? Mechanism of breakdown of food material within the cell to release energy and trapping. Okay. And coming in a while. Give me 2 minutes. And the trapping of this energy for the synthesis of ATP. Now, breaking of CC bonds of complex compounds through oxidation within the cell, leading to the release of considerable amount of energy, is called respiration. Is that clear? So, there are different compounds that can be broken down to release energy. They are known as respiratory compounds, for example, carbohydrates, proteins, fats, organic acids. But carbohydrate is the most often used. Is that clear? So the energy contained in respiratory substrate is not released free into the cell or in single step. It is released in a series of slow stepwise reactions controlled by enzymes, and it is trapped as chemical energy in the form of ATP. ATP acts as the energy currency. Now we know this. Yes. Now coming here, why do plants, do plants also breathe? So they don't breathe exactly, right? There is gaseous exchange. So each living cell, so plants don't have a dedicated breathing system or a respiratory system like nostrils and lungs, all these structures. No. Why? Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. Thick woody stems are not, but then they have some structures like lenticels. Living cells are organized in thin layers inside and beneath the bark. They also have openings called lenticels, told you. Cells in the interior are dead and provide only mechanical strength. Is that clear? Now, talking about glycolysis, this whole pathway was studied by Gustav Emden, Otto Meyerhoff, and J. Paranus. Emden, Meyerhoff, and Paranus. That is why it is known as EMP pathway. In anaerobic organisms, it is the only process in respiration occurs in the cytoplasm, and this glycolysis occurs in all the living organisms, where glucose undergoes partial oxidation to form 2 molecules of pyruvic acid. In plants, this glucose is derived from sucrose. Is that clear? Sucrose will have one molecule of glucose and one of fructose. So sucrose is converted into glucose and fructose by enzyme invertase, also known as sucrase, right? So glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of enzyme hexokinase. So we are actually talking about the first step here. We are talking about the first step here. This is where hexokinase works, right? Now see, glucose 6-phosphate getting isomerized into fructose 6-phosphate. So whenever there is phosphorylation occurring, so glucose changing into glucose 6-phosphate, where will phosphate come from? By utilization of ATP. So one ATP is getting used here. Then when fructose 6-phosphate is changed to fructose 1,6-bisphosphate, another ATP molecule is used here. So we see that these are the two steps where ATP is getting utilized. So you have to know these reactions very carefully. One, glucose to glucose 6-phosphate, and then fructose 6-phosphate to fructose 1,6-bisphosphate, right? After that it changes to G3P, triose phosphate, and here NAD changes into NADH. So the reaction where NADH is produced is G3P to 1,3-bisphosphoglyceric acid. Now see here. When fructose is broken down, it changes into G3P and DHAP. So there is a splitting occurring. So this is a 6-carbon compound, this is 3 and 3 carbon, in total 6. Now this changes to this, they are interconvertible. So whatever is the fate of glyceraldehyde 3-phosphate, the same happens with DHAP as well. So you will count it as twice. Is that clear? So now 1,3-bisphosphoglyceric acid will change to 3-PGA. So here we have 2 phosphates. We are removing 1 phosphate and adding it to ADP. So first ATP molecule is formed here. So here also it happens the same. So 2 ATP molecules created. Similarly, when 3-PGA becomes 2-PGA, this is simple isomerism. 2-PGA becomes 2-PEP, phosphoenol pyruvate. Water molecule is removed, and from phosphoenol pyruvate we'll remove phosphate and keep it as and pyruvic acid is formed. So when 2 phosphate molecules are removed, they are added to ADP. So hence 2 ATP molecules are produced. So why am I saying 2? Because there are 2 PEPs here, right? Yes. So 2 molecules of ATP were utilized, and in return it yielded 4 ATP molecules, right? And 1 NADPH. You must know which step is utilizing ATP, which step is yielding or releasing ATP. Is this point clear? Let's quickly go through this. This in all is a chain of 10 reactions. It is a chain of 10 reactions. Is that clear? So can you calculate how many ATP molecules are directly synthesized in this process? It is saying directly synthesized. What is the answer? 4 ATP molecules are directly synthesized. If it says what is the net gain, then the net gain is only 2 ATP, because for net gain we have to subtract the 2 ATP molecules which were utilized. Are these points clear? So pyruvic acid is the key product of glycolysis. Guys, give me 2 minutes. Let's have a quick water break. And we'll proceed. So it is 1:28. I'll join you in just 2 to 3 minutes. Yes, let's have a quick water break. Eat or drink and just come back in 2 minutes. Yes, guys, are we back? Welcome back. Everybody's here? All right. Let's continue. So we have gone through glycolysis and I've already given you a mnemonic to learn the whole pathway, right? Now see here. So there are three major ways in which different cells will handle pyruvic acid. So there can be three fates. One is lactic acid fermentation, second is alcoholic fermentation, or it can be aerobic fermentation. If it is anaerobic pathway, then either we will get lactic acid or we will get alcohol out of it, depending upon the cell in which that anaerobic fermentation is occurring. Complete oxidation of glucose will give us CO2 and water. For that, it has to adopt the Krebs cycle, which is aerobic respiration. Let's talk more about fermentation. For example, if fermentation, that is anaerobic respiration, occurs in yeast, then we'll get CO2 and ethanol. Now see the two enzymes that are working, because this fermentation occurs in two steps. So basically pyruvic acid will first break down into acetaldehyde, and then acetaldehyde will form ethanol and CO2, right? So the enzymes are pyruvic acid decarboxylase and the second will be alcohol dehydrogenase. These are the two enzymes here, pyruvic acid decarboxylase and alcohol dehydrogenase. While in some bacteria and even in our muscles, when there is less of oxygen, lactic acid is formed from pyruvic acid and the enzyme used is lactate dehydrogenase. Is that clear? So it occurs in muscles also during exercise. Now these are the lines. Not much energy is released and less than 7% of energy in glucose is released, and not all is trapped as high energy bonds of ATP. Now how many ATP synthesized are, and deduct the number of ATP utilized during the glycolysis when one molecule of glucose is fermented to alcohol and lactic acid. So when guys alcohol and lactic acid formation takes place, then we only get two molecules of ATP. In the same way, and those two ATP molecules, net gain of two ATP molecules were had already taken place during the glycolysis. There is no extra ATP produced during the fermentation reaction. Is that clear? So yeast poison themselves to death when the concentration of alcohol reaches about 13%. What is the maximum concentration of alcohol that can be obtained during natural fermentation is just 13%. Now greater, if you want greater concentration, you will have to use the method of distillation. Is that clear? So see this is all glycolysis to pyruvic acid. So pyruvic acid can either form lactic acid or it can form ethanol. Is that clear? Now coming on to aerobic respiration, very very important. Complete oxidation of pyruvate by stepwise removal of hydrogen atoms and leaving 3 molecules of CO2. So how many molecules of CO2 are produced during aerobic respiration? It is 3. Remember this. How and at which step CO2 is released, that also we will take notice of. Now passing on the electrons removed as a part of hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP. First process takes place in the matrix of mitochondria and the second place occurs in the inner membrane of mitochondria. What is the first step that occurs? We are talking about Krebs cycle. The second step that we are talking here is ETS, electron transport chain. Now pyruvate formed by the glycolysis occurs in, where does glycolysis occur? In the cytoplasm. It enters the mitochondrial matrix and undergoes oxidative decarboxylation, which is actually a link reaction. See here, pyruvic acid, coenzyme A, sorry, first is a link reaction. And then after acetyl coenzyme A is formed, that acetyl coenzyme A will enter the Krebs cycle. So pyruvic acid, coenzyme A plus NAD, in the presence of pyruvate dehydrogenase, it will form the acetyl coenzyme A. Now this is the link reaction, guys. This is the link reaction. Is that clear? Now from one pyruvic acid, we get one acetyl coenzyme A and one NADH. But how many pyruvic acids do we have as a result of glycolysis? 2. So 2 acetyl, 2 this, 2 carbon. Is that clear? All these in 2, 2, 2. Are we clear? So 2 NADH are produced during the link reaction itself. Clear? Now during this process, 2 molecules of NADH are produced by metabolism of 2 molecules of pyruvic acid. Just told you. Produced from 1 glucose molecule. Then this acetyl coenzyme A will enter the Krebs cycle. So 1 acetyl enzyme A, 1 Krebs cycle. How many acetyl coenzyme A do we have? We have 2. So from 1 glucose molecule, 2 Krebs cycles will run. See, also known as TCA cycle, starts with the condensation of acetyl group with oxaloacetic acid. So which is the primary acceptor of acetyl coenzyme A? It is OAA. And OAA we know has how many carbons? It has 4 carbons. Acetyl coenzyme A is a 2 carbon compound. This is a 2 carbon compound. This is a 3 carbon compound, right? And together this 2 carbon compound and 4 carbon compound will form 6 carbon compound. So citric acid is a 6 carbon compound. Citric acid is the first stable compound of Krebs cycle, right? Catalyzed by the enzyme citrate synthetase, and a molecule of coenzyme A is released. Are these clear? Then citrate is changing into isocitrate, and isocitrate then forming alpha ketoglutric acid. Let's come down to here. So see, this is the link reaction, this is the link reaction, and then acetyl coenzyme A combining with OAA forming this. So you have to observe that what is the number of carbon atoms present in each molecule. Now see, a simple trick is whenever there is a decline in carbon number, for example, citric acid to isocitric and isocitric to alpha ketoglutric acid. See here, alpha ketoglutric acid has 5 carbon, isocitric and citric has 6 carbon. So there is it has 1 less carbon here in alpha keto. So where is that 1 less carbon gone? It has formed carbon dioxide. Is that clear? So 1 carbon dioxide here, 1 second is here, and then from alpha keto, when it forms this succinic acid, so again there is 1 less carbon atom formed. So this is another carbon dioxide molecule produced. So in total we have 3 CO2 molecules produced after pyruvic acid. That's what it was written. So 1 in the link reaction, 2 during the Krebs cycle. So this is from 1 Krebs cycle, sorry, 1 pyruvate. Is that clear? But how many pyruvates do we have? 2. So in total 6 carbon dioxide molecules will be produced. Please tell me. Are we clear? Right? Now, also see that during which step NADH is getting produced. Which are the reactions which are giving out FADH2? Which reaction gives you GTP? So, all the reactions which are producing, there are 3 steps which are producing NADH. These NADH will later on be oxidized to produce ATP molecule. But ATP is not directly synthesized here. These molecules have to be oxidized. Similarly, FADH has to be oxidized to produce ATP. So the ATP produced by oxidation of these molecules, this is known as oxidative phosphorylation. While when there is direct synthesis of ATP, like from 1,3-bis phosphoglycerate to 3-PGA, so 1 phosphate is adding to ADP, that is substrate level phosphorylation. So you have to know that at which step it is SLP, substrate level phosphorylation occurring, or the oxidative phosphorylation occurring. While this GTP can be regarded as substrate level phosphorylation because it directly gets converted to adenosine by a different metabolic pathway. It doesn't get involved in oxidation or oxidative pathway. Is that clear? So we call it SLP, while these are oxidative. There are 4 steps. 4 steps where oxidative phosphorylation occurring during the fate of pyruvic acid. Is that clear? Now, pyruvic acid. So this is the overall reaction starting from link reaction. This includes link plus Krebs cycle. Link reaction plus Krebs cycle. Pyruvic acid using 4 NAD and 1 FAD, 2 water molecules and ADP in the mitochondrial matrix. It will give rise to 3 CO2, 4 NADH, 4 H positive, 1 FADH2 and ATP. Why 4 NADH? Because 1 was produced during the link reaction also. So in total we have, so this is also in total we have 1, 2, 3 and 4 NADH molecules, only one of FADH2 and one GTP, which is taken as ATP here. Please tell me these points are clear to all. Yes, guys. Are these points clear? Are these points clear to all? Crystal clear, Ananya? Okay. Let's move on to the next. Electron transport system present in inner mitochondrial membrane. So this is how your ETS looks like. First of all, you should know the names of the complexes involved. Complex 1 is NADP dehydrogenase. This is a direct PYQ in match the following. Complex 2 is known as succinate dehydrogenase. Yes, this is dehydrogenase. Then complex 3 is cytochrome BC1, complex 4 is cytochrome C oxidase, and complex 5 is ATP synthase. Now these are the lines. Where is ubiquinone located within the inner membrane? Cytochrome C is a small protein on the outer surface of inner membrane. If you want to better understand this diagram, please refer to marathon in case you haven't. Otherwise, we have discussed it in a far better way. Then complex 4 refers to cytochrome C oxidase containing cytochrome A and A3 and two copper centers. And complex 5 is ATP synthase. This is complex 5 involved in the production of ATP. Now oxidation of one molecule of NAD gives around 2 molecules of ATP. Actually it is 2.5. And from one molecule of FADH, it produces 2 molecules of ATP. Now we are talking about aerobic respiration. What is the role of oxygen? Role of oxygen is limited to the terminal. Terminal means last stage of the process. It's like a showstopper. So oxygen acts as a final hydrogen acceptor. And unlike photophosphorylation, where light is utilized for production of proton gradient, in respiration, energy of oxidation reduction utilized for the same process, and for this reason it is known as oxidative phosphorylation. Is this point clear? So this was now see, this is the structure of ATP synthase. So see, ATP synthase has two components. This is F0 and F1. So this is F0 is the integral membrane protein complex that forms channel protein, transmembrane protein, through which protons can cross the inner membrane, and 4 H positive molecules will pass through the intermembrane space to the matrix down the electrochemical gradient. Focus on this word. It moves down the electron gradient. So higher to lower. Is that point clear? Now see this balance sheet, you should know, we have discussed that. An amphibolic pathway. Why amphibolic? Because it is both catabolic and anabolic. The example is, see, hence fatty acids can be broken down to form acetyl coenzyme A if needed, and the reverse can also happen. That is, acetyl coenzyme A can also form the fatty acids. Hence it is a amphibolic pathway. And then very important table here. So we know these are all energy intermediates. These are substrates, right? Not energy intermediate, sorry. These are the respiratory substrates. So fats broken down to form fatty acids plus glycerol. Fatty acid will ultimately become acetyl coenzyme A. Glycerol will become DHAP and enter the glycolytic pathway and indirectly form acetyl coenzyme A. Then carbohydrates, you know, they form pyruvic acid through glycolytic pathway, and then link reaction produces acetyl coenzyme A. Proteins will first break down to form amino acids, and amino acids will ultimately become pyruvic acid, and pyruvic acid to acetyl coenzyme. So acetyl coenzyme A is the common byproduct, whether it is fat, carbohydrate or protein breakdown taking place. Is that clear? So you have to remember this point. And other than this very obvious question which has already been asked, you should know, how does glycerol enter the pathway? By forming DHAP. How do proteins enter the pathway? By forming the protein, pyruvic acid. Is that clear? Then talking about the respiratory quotient, it is the ratio of volume of CO2 released or evolved upon the volume of oxygen consumed. Now know the values. For carbohydrates, that value is always 1. For fats, it is less than 1. And they have taken an example of tripalmitic acid. So this is carbohydrate. They have taken an example of tripalmitic acid. In case of fatty acids, the value is less than 1, coming out to be 0.7. Okay, so 102 CO2 released and 145 oxygen molecules used. And for proteins, it is again less than 1, but little more than fats, it is 0.9. Pure proteins and fats are never used as respiratory substrates, and also know it for organic acids. Only organic acids will give the value of RQ greater than 1. Is that clear? So guys, with this, we complete the chapter of respiration in plants. Please let me know that these points are clear as well. And now we come on to the last chapter of plant physiology, that is plant growth and development. Let's quickly read this. Yes. So this is the germination. Let's talk about growth. So guys, growth is the irreversible permanent increase in the size of the organ or its parts or even the individual cell, and it always occurs at the expense of energy. So growth is an active process. Now plant growth is generally indeterminate. You can't determine it because it is not fixed. It is not going to stop. It is unlimited because of the unlimited growth. See, because of the presence of meristem at certain locations, it is unlimited growth which will go on throughout their life. Is that clear? Hence it is indeterminate. Now see, you also know that dicot plants and gymnosperms, the lateral meristem, vascular cambium, cork cambium is also present in their life, which all it all contributes towards different types of growths. Increase in girth is known as the secondary growth. Is it clear? So primary growth is occurring at the shoot tip and the root tip and so on. Now growth is measurable. See, at the cellular level, it is measured in terms of increase in the amount of protoplasm, while later on it can be in terms of increase in number. For example, one single maize root apical meristem gives rise to 17,500 new cells per hour, while in watermelon it is in terms of volume increase in size up to 350,000 times. Is it clear? So we have different parameters: increase in cell number, volume, size, length in case of pollen tube, surface area in case of dorsiventral leaf. So these are different parameters. Is that clear? See the phases of growth. So root and shoot apex represent meristematic region. Cells of meristematic region rich in protoplasm, large conspicuous means well developed nucleus, cell walls primary nature and they are very thin, okay, and cellulosic with abundant plasmodesmatal connection. Is that clear? And next to this we have phase of elongation. We've already discussed this in morphology. So phase of elongation and vacuolation, enlargement and new cell wall deposition occurs, and then phase of maturation where maximum size in terms of wall thickening and protoplasmic modifications occur. Then coming on to growth rates, there are two types of growth rates guys, arithmetic and geometric. In arithmetic there is a gradual increase, 1, 2, 3, 4, 5, 6 and so on. And in geometric it doubles up at every step, 1, 2, 2 becomes 4, 4 to 8, 8, 16 and so on. So do you see? While see here it is written, this is zygote dividing. 1 becomes 2, 2 becomes 4, 4 becomes 8 and then, so here we see geometric progression, we see geometric increase in the rate, right, or growth. But later on we see that it changes the pattern and becomes more arithmetic. So remember that stages during embryonic development showing both geometric and arithmetic. So what kind of growth is shown by your zygote or your embryo? It is going to show both arithmetic and embryonic development. Is that clear? Now coming on to this graph. So this graph showing the linear growth. This linear growth indicates the arithmetic growth. What happens? when mitotic cell, in a mitotically dividing cell, only one daughter cell continues to divide, other differentiates and matures. So basically, there is uh, increase of only one cell per division, right? So linear curve, what is the equation? LT is equal to L naught plus RT. You must know what R stands for. It is the growth rate. Now, in geometric growth phase, see here, in geometric, this is geometric, you get a sigmoid curve. In geometric, initial growth is slow, that is the lag phase, then increases rapidly thereafter, which is the exponential or the log phase, and then it again slows down, giving you the stationary phase. So when does a cell show geometric growth? It is when the progeny cells following the mitotic division, both the progeny cells retain the ability. While in linear phase, only one cell will continue to divide, other will go and differentiate. But here, both the cells retain the ability to divide. Hence, but with limited nutrient supply. Why do we get the stationary phase? Because the the availability of nutrients is limited. Limited nutrient supply, growth slows down, leading to the stationary phase. So hence we get S curve or sigmoid. So what is the equation here? It is W1 is equal to W naught E raised to power RT. So E is the base of the natural logarithm, R is the relative growth and also the measure of ability of plant to produce the plant material. Is that clear? So this is the relative growth discussed here. Now another. So when we are quantitatively comparing the growth of the living systems, quantitative growth can be discussed under two categories. One is always the absolute growth rate and second is the relative growth rate. So absolute growth rate is final minus initial. So 10 minus 5, 5. Here 55 minus 50, again 5. So in both the cases, the absolute growth. See, what is the formula for absolute growth rate? Final minus initial. Simple. But then we have second way, which is called the relative growth rate. Now, relative growth rate is indicated by final minus initial upon initial. So, relative is always compared with respect to the initial growth, initial size. Is that clear? So, here the initial is 50 and here the initial is 5. So, the relative growth rates of both the leaves will be different. Is that clear? So, see, for growth of given system per unit time expressed on a common basis, and usually the common basis is initial growth. Is that clear? Now, conditions for growth. What are the external factors and essential factors? See, water, oxygen, nutrients, very essential, right? Turgidity of the cell helps in the extension growth. Linked to the water, water provides medium for enzymatic activities. Oxygen helps in releasing the energy, aerobic respiration. Nutrients, macro as well as micro, required for the synthesis of protoplasm. Is that clear? Now, apart from these, temperature plays a very important role. So you need to provide optimum temperature for the best growth conditions. Then signals like light and gravity will also affect certain phases of growth. Now we know there are these three phenomena occurring: differentiation, de-differentiation, and then we have redifferentiation. What is differentiation? Maturation, where major structural changes both in cell wall and protoplasm occur. For example, to form tertiary element, they lose their protoplasm, become very strong elastic lignocellulosic secondary cell walls to carry the water to the long distances. So this is differentiation. The cell adapts. Cell will show modifications as per the function it has to perform, right? Then at certain points and in certain conditions, what happens? The cells, when they have differentiated, see, when a cell differentiates, it loses the ability to divide. It enters the G0 phase, quiescent phase. It doesn't divide. But in times of need, at certain locations, the differentiated cell can regain the ability to divide under certain conditions. Now, this is called a de-differentiation. So, once differentiated, we call it a permanent cell. And when it regains, we call it a secondary meristem, right? For example, interfascicular cambium, cork cambium, these are all formed by de-differentiation. So these are both examples of secondary tissue. Is that clear? Secondary meristematic tissue. Now, after getting, after dividing, after de-differentiation, after dividing, after fulfilling the need, now these de-differentiated cells can again go back to their mature state. They can differentiate again. That will be known as re-differentiation, where once again they lose the ability to divide and mature to perform a specific function. Are these points clear to all? Please tell me, are these points clear? Now, talking about development, it is a term that includes all the changes that an organism goes through during its life cycle from germination of seed to senescence. Is that clear? All right. Very important, very important. Sequence of development. See, sequences are important. NTA loves sequences. Okay. So, yeah. So, sequence, see the figure. So, meristematic cell, it will show plasmatic growth, as well as it will show expansion. It will undergo differentiation. It will undergo maturation. And after going through all these four processes, this meristematic cell will become differentiated cell or mature cell or permanent cell. Then it will age, show senescence and die. Is that clear? So you have to remember these four processes, and that too in order. What is it? It is plasmatic growth and differentiation, PD, and here it shows EM, PD, EM. Can we remember that? Yes, because sometimes NTA is not really looking for your conceptual knowledge. It is just looking for your rectification capacity. So how much you can rectify, you can just mug up blindly. So even these things can matter. So this is PD, plasmatic growth and differentiation, and then EM, that is expansion and maturation. Is that clear? Now plants follow different pathways in the response to environment or phases of life to form different kinds of structures. This is called plasticity. For example, difference in the leaf structure is called heterophyly. Now we have CLC, cotton, larkspur and coriander. So they show heterophyly according to different stages of life. So in juvenile stage, see the type of leaf, and in adults they have highly dissected leaf. While in case of buttercup, heterophyly is due to environment. In CLC it is because of this life stage, life cycle, and in buttercup it is due to environment. And see here, this is this is a picture. Sorry. Yeah. So this is a picture of larkspur. This is a picture of larkspur, and this is a picture of buttercup. See it in terrestrial habitat, and in water habitat it is highly dissected. Is that clear? Now, development is sum total of growth and differentiation. So there are two phases in development. One is growth, second is differentiation. Now it depends upon two types of factors, intrinsic and extrinsic. Intrinsic includes genetic, that is intracellular, and intercellular factors, which includes PGR, while extrinsic has light, temperature, water, oxygen and nutrition. Is that clear? Today, after completing this class 11 biology NCERT, guys, I will be sharing all these PDFs in my Telegram channel. So in case you have not joined the Telegram channel as of now, so it is Tina Ma'am Biology. So please join this official channel. So, and in this channel only, I am putting all the class notifications as well for the special classes. Please join. I am sending the links for your AIMTs as well. Please take those tests. Is that clear? All right. Now, moving on. Is that clear to all? Please tell me. Yes, guys. Time for the green hearts. Yes. Is it all clear? Is it all clear? Please let me know. Right? Have you all joined? In case anyone hasn't, quickly take a minute and join, because I'll be sharing all these PDFs today in the Telegram channel. All right. Now, moving on to plant growth regulators. So, plant growth regulators are the intercellular factors. Clear? All right. Now, plant growth regulators, small simple molecules. All these, there are 5 different types of PGRs that we are going to study. They can be either inhibitors or they can be promoters, right? So auxins, gibberellins and cytokines are plant growth promoters, while ethylene and ABA, these are the inhibitors. Now these molecules, auxins, gibberellins, cytokinins, ethylene and ABA, they are derived from some different compounds. So see this topic is very, this paragraph is important. So indole compounds, that is indole 3 acetic acid or IAA, derivatives, these are auxins, indole 3 acetic acid or IAA, these are auxins, and what are they derived from? Indole compounds. Then adenine derivatives, for example, N6-furfuryl purpurine, this is a cytokine derivative. Then carotenoids, they produce abscisic acid, terpenes give rise to gibberellic acid, and gaseous is ethylene. Is that clear? Now talking about the discovery of plant growth regulators. So Charles Darwin and his son Francis Darwin, they discovered auxin, did not isolate them. It was isolated by F.W. Wendt. Is that clear? So Darwin and his son, they discovered Darwin, Francis Darwin, Charles Darwin, discovery. Can you remember it like that? So don't get confused. Who discovered auxin? It was Darwin and his son. Who isolated it? It was F.W. Wendt. Then also understand the source. Darwin discovered this auxin from the polyptyles of canary grass using that illumination experiment, right? And the canary grass tip grew towards the source of light, that one, phototropism. And auxin was isolated by F.W. Wendt from the polyptyle of oat seedling, avena seedling, oats, the oats that we eat for very healthy lifestyle. That oat seedling gave F.W. Wendt auxin. Is that clear? Then coming on to, see here. Now, Bacane are the foolish seedling disease of rice seedling by a fungal pathogen Gibberella fujikurae. E. Kurosawa is the name of the scientist. He discovered gibberellic acid from this fungus Gibberella fujikurae. Then F. Skoog and his co-workers, they are known for the discovery of cytokinins, extracts of vascular tissue, yeast and coconut milk. When added along with the auxins, it helped in quick proliferation of callus. So basically, Skoog and his co-workers, they extracted cytokinins from extract of vascular tissue, yeast and coconut milk. Miller et al. Later on crystallized the cytokinin promoting active substances and termed it kinetin. So it was later on crystallized in the form of kinetin. But discovery was done by Skoog from these three tissues. Then we have discovery of abscisic acid by three independent researchers, and they were named differently, inhibitor B, abscission 2 and Dorman. Is that clear? Later on found out to be the same chemical. H.H. Cousins is the name of one person. He discovered this ethylene, a gaseous plant growth regulator. What did he see? He saw that volatile substance released by the ripened oranges hastens, that means quickens the ripening of unripened bananas. Is that clear? Now, talking about auxins, so there are these IAA and indole acetic acid, which has been isolated from the plant. So these are the natural auxins. NAA, naphthalene acetic acid, and 2,4-D, these are the synthetic auxins, first of all. Now, very important and key statements. See, we know that they initiate rooting in stems and applications, and for all that plant propagation, we have read that. Key statement is auxin promotes flowering in pineapple. So flowering is promoted by auxin. In the later part of the chapter, we will see fruit set also in pineapple. Now see, auxins promote flowering in pineapple. Now growing apical bud inhibits the growth of lateral or axillary buds, and this is known as apical dominance. If you want to counteract apical dominance, guys, you need to remove the shoot tips. That is called decapacitation. Decapacitation usually results in the growth of lateral buds. Now this phenomena is used in tea plantation and hedge making. Is that clear? So which which plant growth regulator is used in tea plantation and hedge making? It is auxin. And then we use decapacitation also there. Now, auxins induce parthenocarpy. So parthenocarpy can be natural and it can be induced also because it will give you seedless variety, for example in tomatoes. Then 2,4-D is used as a weedicide or a herbicide to kill only the dicot weeds. It will not destroy the monocot weeds, or if you want to destroy the grass, it will not destroy the grass. Is that clear? Does not mature the affect the mature monocots. So weed free lawns can be obtained by using 2,4-D. Now coming on to gibberellic acid. So gibberellin can cause increase in the length of axis, increase in length of grape stalks, increase in the size of the fruit like apple, improve its shape, delay the senescence, and speed up the malting process in brewing industry. Very important terms. Then sugarcane, it also increases the sugar content, and that is why, see, spraying sugarcane crop with gibberellins increases the length of the stem, also increasing the yield by 20 tons per acre. Spraying juvenile conifers with this, it hastens the maturity period, so you will get the seeds in a lesser time. So for early seed production, promotes the bolting process. What is bolting? Internodal elongation just before the flowering. So as in case of bolting in beetroot, cabbage, and many plants with rosette habitat. Please tell me these points are clear. And then cytokinins, they were discovered as kinetin, told you, modified form of adenine purine from the autoclaved herring sperm DNA. So this is a fish. So it was discovered as kinetin from the herring sperm, and kinetin does not occur naturally in plants. In plants, the form of cytokinin which is found, it is generally this zeatin, isolation of zeatin from corn kernels and coconut milk, right? Now, see, so natural cytokinins are synthesized in the regions with rapid cell division. They overcome apical dominance. So the first antagonistic pair of hormones here is auxins and cytokinins. Auxins promote apical dominance, cytokinins overcome apical dominance, right? They promote nutrient mobilization, which helps in delay of leaf senescence. Talking about this gaseous PGR, so produced by tissues undergoing senescence and ripening fruits, they help in horizontal growth of seedlings and swelling of axis and apical hook formation in the dicot seeds. Respiration rate during ripening of the fruit is also enhanced by ethylene, and this increase in the rate of respiration is known as respiratory climactic. It breaks the seed and the bud dormancy, and germination in peanut seeds and sprouting of potato tubers is initiated. Now see, I told you, flowering is induced by auxins and fruit set in pineapple. In pineapple, flowering is induced by auxin and fruit set is by ethylene. It induces flowering in mango. Flowering in pineapple, auxin. Flowering in mango is ethylene, and fruit set is again in pineapple is by ethylene. Is that clear? Now ethafone is used as a source of ethylene in aqueous solution, hastens the fruit ripening in tomatoes and apples, thinning of cotton, cherry, walnut, and promotes female flowers in cucumbers, thereby increasing the yield. Is that clear? Now talking about ABA. ABA is known as the stress hormone of plants. So whenever there is water stress or any such abiotic stress, it promotes the closure of stomata, increases the tolerance to various kinds of stress. Is that clear? Hence it is a stress hormone. So important role in seed development, maturation, dormancy. Now see, ABA acts as antagonistic to gibberellic acid. So there are two antagonistic pairs of hormones that you have to remember in case of PGR in plants. One is auxin, and it is antagonistic to cytokinins, and second is here abscisic acid to gibberellic acid. Can you remember that? All right. Now, the role of PGR is only one kind of intrinsic control with genomic control. Extrinsic factors could be temperature, light, control plant growth. So, they control plant growth and development through PGR. Please tell me these points are clear to you all. And with this, guys, we complete plant physiology. And now we can start with human physiology. Quickly give me a thumbs up that you're all set, not tired at all. Yes, guys. How many of you? We are going to start with human physiology. Yes, guys. How many of you are ready for human physiology here? So we are starting with breathing and exchange of gases. Right? All right. Let's start with breathing and exchange of gases. So oxygen utilized to break down simple molecules, and we've already discussed this in respiration in plants, right? So process of exchange of O2 from atmosphere with CO2 produced by the cells, this first step of respiration is called breathing. Now see, different respiratory organs, sponges, coelentrates, flatworms, they exchange gases by their body surface using simple diffusion. Moist earthworms use a moist cuticle, insects have tracheal tubes, right? And gills are used by aquatic arthropods or mollusks, and also by fishes. Respiration through gills is known as branchial respiration. Lungs are used by, see, lungs are used by amphibians, reptiles and mammals, and this is known as pulmonary respiration, while frogs can also respire through moist skin, which is called cutaneous respiration. Now, talking about the human respiratory system, starts with the external nostrils. We have a pair of it entering into the nasal chamber and through the nasal passage, then leading into pharynx, which is a common passage for food and air. Then after pharynx, we have it, pharynx opens into the larynx, and larynx will then open into the trachea, right? Larynx is a cartilaginous box and produces sound, hence also known as sound box or the voice box. Now see here, the opening of larynx is guarded by this thin elastic cartilaginous flap called epiglottis, so that food doesn't enter into the larynx or trachea. Is that clear? Now trachea is a straight tube, divides at the level. Which level does it divide at? Fifth thoracic vertebra into right and left pulmonary bronchi. Now see, these bronchi will further divide. Primary bronchi will divide into secondary and tertiary bronchi, and then each tertiary bronchi will further break up into bronchioles, terminal bronchioles. Is that clear? So we have primary, secondary, tertiary bronchi and initial bronchioles. All these are supported by incomplete cartilaginous rings. These are made up of haline cartilage, by the way. They prevent the collapse of these tubules while you are not inhaling. Sorry, while you are inhaling. Is that clear? Now, each terminal bronchiole gives rise to a number of thin vascular bag-like structures called alveoli. So ahead of terminal bronchioles, we have alveoli. Clear? Now lungs, guys, lungs are covered by double layered pleura, and in between we have pleural fluid to prevent the friction, to keep the surface hydrated. Is that clear? So see the structure, this is epiglottis, they have marked, this is larynx, this is all trachea, and trachea is guarded by these cartilaginous rings, right? And these are the initial or the primary bronchi, and each primary bronchi enters the lung. So we have a pair of lungs. You can see all these bronchioles and alveoli filling the lungs. In between the two lungs, we have a space which this is where your heart is exactly placed. Now see, below your lungs, we have this membrane known as diaphragm, characteristic of mammals only, and divides your body into thorax and abdomen also. But primarily here, diaphragm is going to play a very important role in breathing process. Let's see it here. See, external nostrils. So the overall respiratory pathway can be divided into two parts. One is the conducting part, and second is the respiratory or the exchange part. So conducting part starts from external nostrils, goes up to the terminal bronchioles, and then the terminal alveoli and their ducts, alveolar ducts, alveoli and the alveolar ducts, they will form the respiratory or the exchange part. Now, what is the difference? So, as the name says, in alveoli and the alveolar ducts, the exchange of gases will start occurring between alveoli and blood vessels surrounding them. While the conducting part, see, conducting part has these functions. Number one, it will clear the air. You have mucous membrane, you have hair, right? You have mucous lining in the entire pathway. So it is clearing the air from foreign particles. Humidification, it is adding moisture. It brings the air to the body temperature, so thermoregulation. And also right? Right. So these are the functions done by conducting part. Number one, air clearing, humidification and thermoregulation, while exchange part is the actual site for gaseous exchange. Lungs are present in an airtight chamber which is dorsally means on the back side, dorsally formed by vertebral column. Ventrally there is a sternum, on the sides literally it has ribs, and on the lower side it has this dome shaped diaphragm. Is that clear? Now this arrangement is very important for breathing. We'll see. So this was also a PYQ. We got this question this year. So respiration involves which of the following steps? So breathing or the pulmonary ventilation, exchange between atmosphere and lungs, followed by diffusion of gases across the alveolar membrane and the blood, then transport of gases by the blood and then between blood and tissues, and finally utilization of oxygen by the tissues to produce energy. This is called cellular respiration, which has already been discussed. Now talking about mechanism of breathing, we have two phases: inspiration, inhalation; expiration, exhalation, right? So inspiration can occur if pressure within the lungs, that is intrapulmonary pressure, is less than the atmospheric pressure. See, that's a basic physics. Air will always move from high pressure to low pressure area. You want air to go inside the lungs, there has to be low pressure inside your lungs. You want the air to come out, you have to create higher pressure area inside the lungs. Now you cannot directly manipulate the pressure. What you can do is you can manipulate the volume. Let's do that. So expiration is when intrapulmonary pressure is higher than the atmosphere. So for all these things to happen, we have these structures. We have diaphragm, specialized set of muscles, external and internal intercostal muscles, ribs. They are generating these differences, right? Now inspiration is initiated when contraction of diaphragm increases the volume of thoracic chamber in the anterior posterior axis, and contraction of external intercostal muscle lifts the ribs. Do you see it here? And sternum causing the increase in the volume of the thoracic. So when diaphragm contracts, it becomes flattened. So there is increase in the volume in anterior posterior axis. When your external intercostal muscles contract, your ribs are raised, your sternum is raised. So the volume is increasing in dorsoventral axis. Are you getting that? So overall, when the volume increases, increase in the volume decreases the pressure. Pressure and volume are inversely related, right? Is that clear? Now the reverse happen in case of expiration. When ribs and sternum, they return to the original position, diaphragm also becomes dome shaped. So decrease in volume will increase the intrapulmonary pressure and hence air will move out. Now what is the breathing rate? Not talking about heart rate. Breathing rate. How many times do you breathe in 1 minute? 12 to 16 times. Volume of air involved in breathing movements. Now that can be measured by a spirometer and it has a clinical significance. It helps to understand your lung health. Now talking about the respiratory volumes and capacity. First of all, volume is always a single value and capacities are always more than one values. So tidal volume during normal inspiration. You inhale, you exhale. How much? Only 500 ml. So per minute it becomes 6,000 to 8,000 because 500 into 12, 500 into 16. So that's how you calculate. Now inspiratory reserve volume. This is during forcible inspiration. You inhale forcibly, but remember you exhale normally here. The value is 2,500 to 3,000 ml. In expiratory volume, there is forcible expiration. You exhale forcefully, but inspiration is normal. What is the value? That is 1,000 to 1,100 ml. Now, residual volume, no matter how strongly, how forcibly you exhale, you can only exhale to a certain amount. Some amount will remain in lungs even after forcible expiration. This is 1,100 to 1,200. This is called residual volume. Now, these are the 4 volumes. Is that clear? Now, talking about the inspiratory capacity. What is the total volume of air that a person can inspire after a normal expiration? Exhaled normally, now inhaled, tidal volume and extra. So total is TV plus IRV is inspiratory capacity. Similarly, EC is TV plus ERV. Forcible exhaled, right? After normal expiration. Normal expiration, person exhales forcefully. Now, we have FRC, functional residual capacity. That is volume of air that remains in lungs after normal expiration. Is that clear? So it includes ERV, which can be forcefully thrown out, and then we have RV, which can never come out. And then vital capacity, volume of air person can breathe in after forced expiration. So maximum means forced. Is that clear? So actually in vital capacity, both the directions there have to be forceful things. Forceful inhalation after forceful exhalation, or forceful exhalation after forceful inhalation. Both ways it has to be maximum. So it includes ERV, TV and IRV. There are three components. And then total lung capacity. It includes total volume accommodated in lungs at the end of forced inspiration. So RV, ERV, TV and IRV. Or if I just combine these three, ERV, TV and IRV, it becomes vital capacity, or simply vital capacity plus residual volume. So these formulae will be utilized in solving the numericals if you get any. Is that clear? Is that clear? Please let me know. Yes guys, quickly let me know that all these points are clear. And guys, kindly click on this link given in the chat. It is already pinned as well. So this is for UAITS, India's biggest test series, only for ₹1. I've told you already. So you have to take these tests, guys. You have to take these tests. So this is Unacademy All India Test Series just for ₹1, where you will get 10 minor tests, 3 major tests, 6 subject-wise tests and 6 tests here. So you are getting 19 and 6, 25 tests here just for ₹1. And it is not about just like 25 tests. You have how many days left? It is already 10th. We are just left with 11 days. Rather, today onwards, you should be solving 2 papers every day. One, the AIMT, that is on Wednesday, Friday and Sunday, just thrice a week. Otherwise, take 2 tests every day. Solve all these tests. Who will stop you from cracking your NEET if you're taking all these tests every day? And that too at your convenient time. But my strong suggestion is take it between 2 to 5. Is that clear? So then moving on to, so practice all these things that we are studying. How are you just studying blindly? I've seen many students studying blindly and asking silly questions. Our rank is not improving. Our marks are not improving. How will your marks improve if you're not practicing more questions? And just practicing is not enough. You have to analyze also. A proper analysis will take around 1 hour, guys. So video solutions are also provided, where educators are telling you that how to think on a particular question. What are the keywords that you need to concentrate on? Please solve those questions and practice questions with me also in the special class tonight as well. Now, exchange of gases, alveoli, primary site of exchange of gases, simple diffusion occurs. Solubility of gases will depend upon this diffusion will depend upon the solubility of gases as well as the thickness of the membrane. So these are the partial pressures, because air will move from one place to another according to the pressure difference. And because air is a mixture, so every gas is contributing partially. So we use the term partial pressure. So see the partial pressure of oxygen in atmosphere 159, in alveoli 104, in deoxygenated blood 40, in oxygenated 45, and in tissue again 40. Sorry, in oxygenated blood it is 95. I'm talking about oxygen. In CO2, atmosphere has 0.3 mm of mercury, alveoli has 40, deoxygenated will have higher value 45, and blood will have 40, and tissues will again have 45. Now these values can change depending upon the metabolic activity of tissues though, but rest remains the same. Now see here. Pressure of oxygen 104 in the alveoli, and we said in deoxygenated blood it is 40. So oxygen will move from alveoli into the blood, and the deoxygenated blood will become oxygenated. Yes? And then CO2 45 here, 40 here. So carbon dioxide will move into the alveoli and go out. So this is the oxygenated blood. See, pressure 95 and 40. And here pulmonary artery and systemic veins carrying deoxygenated blood. What is the value? And that is how exchange is taking place. Now see here. Solubility of CO2 is 20 to 25 times higher than that of oxygen. Is that clear? Now in case, guys, if you feel I'm not reading entire NCERT and not reading all the words given here, please understand, you also don't have to do that now. Have that faith, you remember things already, you have good memory. If you'll, always stay in that panic mode, okay, if I forget, you'll definitely forget. Take it for granted. Whatever you are thinking, it is going to happen. So don't keep on thinking negative, that okay, what if I forget, let me read it again. No, not required. Solve more questions. You remember things, you will always remember things. Is that clear? Don't stay in that panic mode. See, little bit of stress is healthy because it keeps you going, but that stress also has to be regulated. Don't let it go out of the boundary and give you panic attacks. Panic won't help. Is that clear? So just solve more questions. Solubility of CO2 is 25 times higher than that of oxygen. What is the diffusion membrane made up of? Three things. Squamous epithelium of the alveolar wall, then basement membrane, and the endothelium of the blood capillary. These three layers make the diffusion membrane. And what is the total thickness of all the three? Less than 1 millimeter. Is that clear? Now, moving on to transport of gases. For oxygen, 97% of oxygen transported by hemoglobin, 3% through plasma. CO2, 20 to 25% by RBCs, that is by hemoglobin forming aminohemoglobin, 70% as bicarbonate, and 7% in the dissolved state through plasma. Now, transport of oxygen. Hemoglobin is the major carrier, red colored iron containing pigment, and it binds with oxygen in the reversible manner. So, reversible manner means if oxygen can bind, it can unbind, it can dissociate as well. Now, when we study the binding between oxygen and hemoglobin and the percentage saturation of hemoglobin, we get a sigmoid curve again. Partial pressure along x-axis and percentage saturation along y-axis here. So sigmoid curve again, we have lag phase, we have log phase, and we have stationary phase. And one hemoglobin can carry maximum of 4 oxygen molecules. So this association is called oxygen dissociation curve, ODC. Now, what are the conditions which will help oxygen and hemoglobin association or binding of hemoglobin and oxygen? See, high partial pressure of oxygen, low partial pressure of CO2, very important. Less H positive concentration and low temperature. So, they favor the formation of oxyhemoglobin. While in tissue side, what will happen? There is less pressure of partial pressure of oxygen, high partial pressure of CO2, high H positive and high temperature. So this will favor the dissociation. Is that clear? So all these factors that I have written here, all these factors, they will cause the left hand shift like this. While these factors, they will take a right hand shift. They will cause a, they will cause a right hand shift. Is that clear? So right hand shift will indicate, by the way, this is going to wait. Yeah. So this is going to be like this and this is going to be like this. Is that clear? So note down the factors which will cause left hand shift or association and right hand shift, that is dissociation. Now, moving on to this very important line, every 100 ml of blood will transport just 5 ml of oxygen under normal physiological conditions. But if the demand increases, this value will also alter. Now, transport of CO2, 25%, 20 to 25% is with hemoglobin forming carb-aminohemoglobin. Now, oxygen is a major effect that affects this binding of CO2 and hemoglobin. Now, RBCs contain very high concentration of enzyme, carbonic anhydrase, and minute quantities in plasma too. So, this reaction, CO2 plus water in the presence of Ca forming carbonic acid, forming and dissociating into bicarbonate and H positive ions. This is occurring in your RBC. Same reaction is occurring outside also in the plasma, because the same components and same enzyme is there. But in plasma, we have it in minute quantities. So what will happen? Outside in the plasma also, these bicarbonate ions are formed. But here we have high concentration of bicarbonate ions. Here we have low bicarbonate ion concentration. What will happen? They'll start moving out. And now these bicarbonate ions will react with sodium and potassium to form sodium bicarbonate and potassium bicarbonate, forming, they will be carrying carbon dioxide. So maximum carbon dioxide transport occurs in the form of bicarbonate ions. Now when bicarbonate ions were moving out, it was compensated by chloride ions. It was compensated by chloride ions. Is that clear? Now, that is called the chloride shift. So at alveolar site where partial pressure of CO2 is low, the reaction proceeds in the opposite direction, right? Now every 100 ml of blood will take only 4 ml of CO2 to the alveoli, but this is under normal conditions. Then regulation of respiration. See, it is your brain taking control now. There are two parts of the brain which are involved. One is the medullary region and second is the pons varolai. Is that clear? So in medulla we have respiratory rhythm center, and in pons, guys, we have pneumotaxic center, PP, right? Now pons varolai or pneumotaxic center, it reduces the duration of inspiration. So this is a PYQ. The more impulse that you get from pneumotaxic center of pons varolai, the duration of inspiration goes on decreasing. So it will make it a shallow breathing basically, right? So chemosynthetic sensitive area adjacent to the rhythm center here, which is highly sensitive to carbon dioxide and hydrogen ions. Now receptors are associated in receptors for the CO2 and H positive ion. They are present, one in aortic arch and second in the carotid artery, the one which carries blood to the brain, right? So they recognize changes in CO2 and H positive concentration, send signal to this respiratory rhythm center, and this will in turn determine that more breathing should occur or less breathing should occur. Is that clear? Very important line. Role of oxygen in regulation is quite insignificant. So the fact says if you have more CO2, there will be more breathing rate. More H positive ions, more breathing rate, and the vice versa. So oxygen plays less significant role. Talking about the disorders, guys, asthma, difficulty in breathing. Characteristic sound is wheezing sound, and it is due to inflammation of bronchi and bronchioles. Emphysema, chronic disorder, alveolar walls damage, respiratory surface decreases because alveolar walls are damaging, and the main cause is cigarette smoking. And then we have occupational respiratory disorders. This is because of certain occupations, certainly not the white collar jobs, but those who are working in, who are directly exposed to these components like grinding, stone breaking, dust. Body cannot cope up with it fully, and long exposure gives rise to inflammation, causes fibrosis and serious lung damage. So that is why people who are directly in contact with these microfibers, micro elements, particles, they should be given protective masks as per the Factory Act. Is that clear? So this is breathing and exchange of gases. So these are the important lines that we have read from this chapter. Is that clear? Now moving on to the next is body fluids and circulation. Yes, guys. Let's move on to the next chapter, body fluids and circulation. Let's talk about the lower organisms. So sponges, coelentrates, they circulate water from the surrounding through body cavities to facilitate their cells. Do you remember in sponges we have spongocoel? We've read that. Coelentrata have gastrovascular cavity, right? Then blood is the most commonly used body fluid for most of the higher organisms. Lower organisms don't have that. Another body fluid is lymph. So one is blood and second is lymph. Blood and second is lymph. Now talking about blood first, it is a special connective tissue consisting of the liquid part known as plasma and the cellular part, cells, are together known as formed elements. So plasma, slight yellowish, straw colored, constitutes 55% of the total blood. 90 to 92% of plasma is water and 6 to 8% is proteins. And what are these proteins? These proteins are fibrinogen, globulins, and albumins. These are the three major proteins present in blood. Fibrinogen, as we know, is involved in clotting. Globulins, like immunoglobulins, they are the defense providing antibodies, right? And then albumins, the smallest of them all, they help in maintaining the osmotic balance of blood. So how much blood will be present, how much water will be present in blood, it will be all determined by albumins. Is that clear? Now, it contains minerals also. Factors for coagulation or clotting are also present in blood in the inactive form. So if you minus these clotting factors from plasma, you get serum. So plasma minus clotting factors is serum. Is that clear? Let's talk about the second component of blood, that is formed elements. And these formed elements constitute 45% of the total blood. Collective term used for RBCs, erythrocytes, WBCs, leukocytes, and thrombocytes, that is platelets. RBCs, the most abundant of them all. So if you have to arrange these formed elements in the decreasing amount, so the maximum are RBCs, then it is platelets, and the least ones are the WBCs, right? So how many RBCs do you have? It is 5 million to adult man, actually 5.5 million. In females, we have little less, but we can count it at almost the same. So RBCs are formed in red bone marrow. So mature RBCs are devoid of nucleus. Is that clear? Biconcave in shape, they carry hemoglobin. Why do they lose nucleus in even other organelles like mitochondria, ER, GB? They don't have all these. Why? To increase the surface area to carry maximum of hemoglobin. Now, healthy individual has 12 to 16 grams of hemoglobin per 100 ml of blood. Is that clear? See the units also. It is gram, not microgram. Now, what is the average lifespan? That is 120 days, after which it will be buried in the graveyard, that is spleen. By the way, spleen will also act as an emergency blood bank as per the requirement. Talking about leukocyte or WBCs, they lack hemoglobin, lesser in number. What is the number? 6,000 to 8,000 per millimeter cube. Very short-lived because they are soldiers, right? Now we have 5 different types of WBCs. See here, neutrophils, basophils, eosinophils. These 3 are the granulocytes. They contain granules of different nature. And the lymphocytes and monocytes, these are agranulocytes. They do not have granules. So all have their specific functions. By the way, neutrophils are the most abundant and they are phagocytic. Basophils are the least and they are in 0.5 to 1%. How do we remember? Remember it as null map. What do you remember? Null map. So null map. So this is maximum and basophils are minimum. How much are the neutrophils? 60 to 65. How much are basophils? 0.5 to 1%. Is that clear? Together, then monocytes. Monocytes, it is written 6 to 8%. They are also phagocytic, just like neutrophils. Basophils, I've told you 0.5 to 1%. They are secretory. They secrete 3 things. See, histamine, which causes inflammation. Serotonin, which does not cause, rather it antagonizes that. And heparin is your anticoagulant, right? Eosinophils are 2 to 3%, right? So this is 60 to 65%. This is done. 0.5 to 1%. Is that clear? And eosinophils 2 to 3%, they have important role to resist infections and during allergic reactions. And how many lymphocytes do you have? 20 to 25%. Null map. So we know lymphocytes are of 2 types, B and T. Clear to all? Then coming on to platelets, also known as thrombocytes. Now, platelets are not real cells. They are actually cell fragments. They are derived from their mother cell known as megakaryocyte. The name itself says large nucleus. But while megakaryocyte changes into platelets, they become like cell fragments, irregular in shape, very sticky, right? Now, this structural change in the platelets helps them to form a good clot. Irregular boundaries will fit into each other like this, right? And they will close the bleeding area. So what is the number? That is 1.5 lakh to 3.5 lakh platelets per millimeter cube. And what is the function? Helps in coagulation. Is that clear? Now, coming on to blood groups. So blood group depends upon the antigen. See, blood group is exactly equal to, blood group is the same as antigen on the RBC surface. Depending upon the antigen, it is your blood group. See, depends on the surface antigens. Antibodies, that is proteins produced in response to antigen, antibodies are present in the plasma. Antibodies are present in the plasma. These are present on the RBC surface. So this is the group of blood donors and recipients. See, antigen and blood group, they are just the same. Is that clear? Antibodies are against it. So A will have anti-B, B will have anti-A, AB will have no antibody. That is why it will be known as a universal recipient. And O will have no antigen. That is why they are the universal donors. Is that clear? So you should be careful about who can donate and who can receive blood. Apart from A, B, there are other antigens also present on RBC surface. For example, RH. Now this was first discovered in rhesus monkey, and that's how the word RH came up, right? There RH. Now, according to RH, we have the special case of RH incompatibility. Now, RH incompatibility arises when RH negative mother, understand this? RH negative mother marries RH positive father, because this will lead to RH positive baby. Now, this leads to the special condition called as erythroblastosis fetalis, right? This is RH incompatibility. What happens? Now, just look at the mother. RH negative, baby is RH positive. So, first pregnancy would be normal, but RH antigens of the fetus do not get exposed to RH negative blood of the mother in the first pregnancy, so it is normal, as the two bloods are well separated. But during the delivery, during the pregnancy, nothing happens. But during the delivery of the first baby, there is a possibility of exposure of maternal blood, this negative blood, to the small amount of RH positive blood of the fetus. Now, what will happen? Now, mother's body will start preparing antibodies against RH. Now, these antibodies won't do any harm during the delivery. They will harm the next pregnancy, right? So, what will happen in subsequent pregnancies? RH negative mother, the antibodies leak into the blood of the fetus. Is that clear? And destroy the fetal RBCs, causing severe anemia, severe jaundice, and the baby will die. Stillbirth would happen. This is called erythroblastosis fetalis. Now, this can be avoided by administering the anti-RH antibodies to the mother immediately after the delivery of the first child. Is that clear? Next is coagulation of blood. Now, for coagulation, that is the coagulum or the clot formation, network of threads, fibrins are involved. Fibrins will entrap the dead and damaged formed elements, RBCs and WBCs, right? Now, basically, your blood has fibrin in the inactive form as fibrinogen, right? Now, this fibrinogen becomes fibrin. Why? Because of enzyme thrombin. Now, you have thrombin also, but in inactive form, that is prothrombin. So, prothrombin will become thrombin. This thrombin will change fibrinogen into fibrin. Is that clear? And why does prothrombin change into thrombin? Because platelets will release this enzyme called thrombokinase. So it is a cascade reaction. One thing follows the other automatically. All you need is the platelet to get rupture and release this thrombokinase. Rest, everything will follow. So this is how clot formation is going to take place. And calcium ions are important for clotting mechanism. Now, talking about another body fluid, that is tissue fluid or lymph. This fluid is released out in the interstitial fluid or the tissue fluid, helps in the exchange of nutrients and gases. So, it is a colorless fluid containing specialized lymphocytes responsible for immune response. Important carrier of nutrients, hormones, and fats are first absorbed in these lymph in the lacteals present in the intestinal villi. What are lacteals? Lacteals are basically lymphatic capillaries. So the lymphatic capillaries below the intestinal villi are given the specific name lacteals. Is it clear? So fats are first absorbed in lacteals, and then when the lymph will flow back into the blood, fat will be put into the blood. It doesn't directly enter your bloodstream. Talking about the circulatory pathway, we already discussed in animal kingdom. We have open and closed. So by the way, closed is only found in annelids and arthropods, while open is found in rest of them. All those which have well-developed circulatory system other than annelids and chordates will have open circulatory system. And you know what is the difference? In open, we don't have capillaries, so blood fills the whole space. And enclosed, there are well developed channels, that is blood will only flow through those channels, arteries, capillaries, veins only, right? Now all vertebrates possess the muscular heart. Fishes have two chambered heart, amphibians and reptiles will have three chambered heart, except crocodile family, right, which will have four chambered heart. Crocodiles, birds, mammals will have four chambered heart. In fishes, blood pumps out of the heart only once, so there is single circulation, while in amphibians and reptiles there is incomplete double circulation, and in birds and mammals we have complete double circulation. There is complete double circulation. Are these clear? Now let's talk about human circulatory system. So human circulatory system, like blood vascular system, consists of heart, blood vessels and blood. We have read about blood already. Let's talk about heart. So overall circulatory system is majorly contributed by mesoderm, and so is your heart, right? Present between the two lungs, slightly tilted towards the left. Now this explains why we feel our heartbeat towards the left side. The covering of the heart is pericardium. Again, this is also double layered, just like pleura, and in between the two pericardial layers we have pericardial fluid. Now because humans have four chambered heart, the upper small chambers are called atria, the lower large ventricles chambers are known as ventricles. Now see, the right atrium and right ventricle, the right side, it has a valve, three muscular flaps called the tricuspid valve, while the bicuspid valve is present between the left auricle and the left ventricle. This is the structure of heart. I hope you can easily identify these structures. Now the valves, what is the function of valves? Whatever the valves be, wherever the valves be, they always prevent any backward flow. Now, entire heart is made up of the cardiac muscles. We have read through the structure. Now, specialized cardiac musculature called the nodal tissue. Now, I want to mention a PYQ here. See, all these structures that you are going to talk about, SAN or AVN or AV bundle of Purkinje fibers, they are conducting the impulse from one area to another and helping in the proper synchronized movement of heart. But remember, none of them is a nerve fiber. They are all modified cardiac muscle fibers. Muscles can also conduct impulse, right? So they are all modified, see, specialized cardiac musculature called the nodal fiber. And present in the right upper corner of the right atrium is a SAN. In the lower left corner we have of the right atrium we have AVN, then we have AV bundle, then atrioventricular septum, AV bundle is present in the atrioventricular septum, and then it breaks down into the Purkinje fibers. So this is the order of sequence also along which the impulse flows. Now nodal musculature, we are talking about SAN. So SAN has the ability to generate the action potential automatically. So hence it is auto-excitable. And because SAN is present within the heart, this makes our heart myogenic. So the impulse for the heartbeat that generates within the heart. There is no external impulse coming from anywhere else, right? So our heart is called myogenic. Now see, SAN can generate the maximum number of 70 to 75 minutes rhythms per minute, potentials per minute, and it is responsible for initiating and maintaining the rhythmic contractile activity. Hence, it is known as a pacemaker. Average is 72. And by chance, if your heartbeat goes faster, it means SAN is producing more impulse. So, the number of impulses of potential produced by SAN is exactly equal to your heart rate. Is that clear? Alright, now talking about the cardiac cycle. See, all four chambers relax. This is called joint diastole. What happens during joint diastole? TV and BV open. Blood from the pulmonary veins and vena cava flowing into the right and the left atria, and because TVBV are open, it is flowing down into the ventricles. SLV is closed at this hour. Now, at the end of, now this joint diastole goes for 0.4 seconds, right? This is joint diastole. At the end of 0.4 seconds, SAN is going to generate an impulse. As the SAN generates impulse, both the atria will contract, and this will stay for just 0.1 second. Now, what will happen during 0.1 second? The blood which is coming into the atria, right and left both, it will be pushed down into the ventricles. So there will be fast delivery of the blood. So blood enters into the ventricles. 30% of the ventricle filling will occur here, while 70% has already taken place during the joint diastole. Is that clear? Now see, now after 0.1 seconds, impulse reaches the ventricles and ventricular systole begins, where while atria undergoes relaxation. So ventricular systole and atrial diastole, they go hand in hand. They are overlapping each other. Is that clear? So during the ventricular systole, first of all, TV and BV will close, and this will produce the first heart sound that is known as love. So any heart sound is produced, be it love or dub, it is produced because of closure of valves. And note down, when did it occur? It occurred during the ventricular systole. During the beginning of ventricular systole, we got the first heart sound due to closure of TV and BV. Is that clear? Now, ventricular pressure will increase and semilunar valves will be forced open. These are all sequences. Right? So forced open, ventricles will now relax, blood will flow out, and the ventricle blood flows out. Ventricles now relax. This is called ventricular diastole. Closure of semicircular valves will occur, leading to the second sound, that is dub, during the end of ventricular systole. So both the sounds are produced during the ventricular systole only. Please take down this point. Is that clear? One, lub during the beginning and dub during the end of it. Please tell me these points are clear. Yes? Yes, guys, please let me know that all these points are clear so far. All these points are clear? Superb. Now, TV and BV will be opened. When SLV will close, TV, BV will open, and now ventricles and atria are now again in the relaxed state. Is that clear? Now, this sequential event in the heart, which is cyclically repeating itself, is called the cardiac cycle. Heartbeats are 72 times. So, the duration of cardiac cycle will come out to be 0.8 seconds. Each ventricle. See, remember, the amount of blood entering both the both the atria is equal. Reaching both the ventricles is equal. Getting pumped out of both the ventricles is equal. Okay. Now see, what does it say? Yes, each ventricle pumps out approximately 70 ml of blood, which is called the stroke volume. Stroke volume multiplied by heart rate gives you the cardiac output. Cardiac output is how much volume of blood pumped out by each ventricle per minute, because we are taking heart rate into consideration. So average cardiac output is 5,000, that is 5 liters in a healthy individual, which is almost the total amount of blood present. Is that clear? Now, see here. So the cardiac output of the athlete will be much higher than that of the ordinary man. How do you measure your heart rate? That is by stethoscope. First sound lub, by closure of the second sound dub, that is by closure of SLV, and these sounds are of clinical significance. Now let's talk about ECG. In case you are using your old NCERT, the full form for ECG was electrocardiograph. No, it has been rectified to electrocardiogram. First, take notice of this, and then ECG, the machine used, the device used here is electrocardiograph. So see the difference. Gram is that graphical representation. Device is, or the machine is graph. Remember the opposite. And so ECG is a graphical representation of electrical activity of heart. It is not the mechanical activity, whether it is contracting or relaxing. No, it is the electrical activity. And mind you, electrical activity will be followed by the mechanical activity. So first you will get the P wave or the QRS waves and so on. And then P wave will be followed by the atrial contraction. Is it clear? So we will read through these lines. See here. So while you have to take ECG of an individual, we need at least 3 electrical leads, one to each wrist, right and left, and then to the left ankle, but the number of leads can be increased as per the requirement. Clear? This is a typical ECG, P, QRS and T wave, right? Now the P wave represents the excitation or depolarization of atria, which leads to contraction. So P wave is not atrial contraction, it will lead to, because this is the electrical activity. Once that electrical impulse arises, it will lead to the contraction or the movement. Is that clear? Now QRS complex, it represents a depolarization of ventricles, and then contraction starts shortly after Q wave. I hope you understand this point now. And then we have T wave. T wave represents the return of ventricles to the normal state, that is repolarization, and marks the end of systole. Counting the number of QRS complexes gives you the number of heart rates. So number of potentials, number of action potentials generated by SAN is equal to heart rate, and it is also equal to the number of QRS complexes. So depends on how you are measuring it. Is that clear? Now see, deviation from this typical shape indicates an abnormality of the disease and hence has a clinical significance. Let's talk about double circulation. Now we know that blood enters the heart twice. First in the form of deoxygenated, goes to the lungs, gets oxygenated, comes back to the heart, and heart will pump that oxygenated blood back to the body. So the same blood is entering the heart twice. First in the deoxygenated form, then in the oxygenated form. See, and it flows through the blood vessels. So we have 3 types of blood vessels: arteries, veins and capillaries, right? Arteries and veins consist of 3 layers. The inner layer is made up of squamous epithelium, middle layer tunica intima, inner layer is tunica intima, middle layer is made up of smooth muscles and known as tunica media. Also it contains elastic fibers because it has to show vasodilation and contraction, right? Then the outer layer is made up of fibrous connective tissue with collagen fibers. This is known as tunica externa. Now the difference between arteries and veins is only with respect to tunica media. Artery has a thicker media and vein has a thinner media. Also in veins there will be valves present to prevent the backflow. Arteries won't have the valves. So it is thinner in veins. Is that clear? Now, all right, talking about the double circulation. So the overall circulation can be divided into 2 parts: pulmonary, in which heart and lungs are involved. So blood from heart goes to the lungs, comes back to the heart. That is from RV to lungs and back to LA, right? And then the part of circulation between body and heart, that is from left ventricle to the whole body, getting providing oxygen, coming back to the right auricle. That is your systemic circulation. Is that clear? Now there is a special pathway between digestive tract and liver, which is known as hepatic portal system. It carries blood from intestine to liver before it is delivered to the systemic circulation, and this is very important for storage of the nutrients by liver, right? And then we also have coronary circulation. It is between blood and the cardiac musculature. Is that clear? Now regulation of cardiac activity, again, normally the activities are controlled intrinsically because the initiator, pacemaker, is within the heart, right? So it is auto-regulated and heart is called myogenic, already told you. But your brain can influence it. Brain cannot start it. Brain can influence it. Medulla oblongata can moderate the cardiac activity through autonomic nervous system. See, or by neural signals are sent through sympathetic nervous system and parasympathetic. Right now, during normal conditions, it is parasympathetic, and during your NEET exam, it will be your sympathetic nervous system controlling your heart rate, right? Are these points clear? Now see, so parasympathetic will decrease the heart rate, speed of conduction, and thereby also decrease the cardiac output, while adrenal medullary hormones, because adrenaline works in tandem with sympathetic nervous system, right? It will increase the heart rate, increase the speed of conduction, and also increase the cardiac output. Let's talk about the disorders here. So high blood pressure, hypertension. See, normal blood pressure is 120/80. 120 is the systolic pressure, 80 is the diastolic pressure, right? But if the pressure remains above 140/90 continuously, then person is suffering from high blood pressure, and it can lead to heart diseases and many vital organ diseases to brain and kidney. Talking about coronary artery disease, so this occurs because of the cholesterol deposition in coronary artery, which is known as atherosclerosis, deposits of calcium, fat, cholesterol and fibrous tissue, and makes the lumen of artery narrower. So it can also lead to blockage. It can lead to heart attack, myocardial infarction, right? What is angina? So angina pectoris is the acute chest pain when not enough oxygen is reaching the heart muscle. And why does it occur? It can be because of coronary artery disease, right? It is more common in any age, but more common among the middle-aged and elderly due to conditions that affect the blood flow. Is that clear? Now, last not the least, we have heart failure. See, when do we fail? When we are not performing to our optimum, right? Then heart failure is when not pumping blood effectively to meet all the needs. Clear? And also known as congestive heart failure because of congestion of the lungs. Then we have cardiac arrest. When you arrest someone, you stop someone. So basically the heart stops beating. Maybe your sand did not produce impulse. So it results in sudden collapse of the person. Heart attack, when heart muscle is suddenly damaged by inadequate blood supply. Again, it must occur because of CAD, coronary artery disease. Now please tell me all these points are clear to you all. So with this, guys, we complete second chapter of human physiology, and now moving on to excretory products and elimination. Yes, guys, time for thumbs up. Please let me know that all these two chapters or all the chapters that we have done so far are completely clear to you all. Yes, please. Crystal clear, Ananya? Have we all joined the Telegram group? It will take me another hour or so, and we will complete the syllabus of class 11th NCERT. Yes? All right. Is there any doubt? Is there any doubt in the session so far? Please let me know. No, ma'am, Ananya? Why not? I've already mentioned multiple times. See here. So that is Tina, ma'am, biology. I'll be sharing all the PDFs in this Telegram channel. All the PDFs. Even of the previous marathons I have shared here. It have all the important points highlighted. Rest lines we skip. Kumar, Saurabh, rest, as I've told you, we already remember. Please have faith in yourself. You remember other lines. You know what is there. These are the lines from which maximum questions have come up or will come up. They include PYQs, they include expected questions, and they include all the concept-based lines. Rest lines, yes, you can skip. Is that clear? All right. Let's start with excretory products and the elimination, guys. Ammonia is the most toxic form and uric acid is the least toxic form. So urea remains the moderate form. So many bony fishes, aquatic amphibians, aquatic insects, bony fishes, they are ammonotelic, and they excrete this ammonia by diffusion through body surface or through gill surface. Is that clear? Just a second, guys. All right, guys. NCERT practice so that any question we tackle which is best. Yes. All right. So next is uric acid for conservation of water, right? Mammals. So terrestrial adaptation is to produce uric acid instead of ammonia. So mammals, many terrestrial amphibians, marine fishes mainly excrete urea and are called ureotelic. Is that clear? Reptiles, birds, land snails and insects, they release uric acid in the form of pellet or paste to minimize the loss of water. All these lines are important. Press. Then invertebrates, they have simple tubular forms to remove their excretory waste, and vertebrates have a complex tubular organs called kidneys. Antennary glands. See, yesterday when we were discussing animal kingdom, I told you in the chapter of animal kingdom it is just written that arthropods excrete through Malpighian tubules, right? But not all do. See, antennary glands or green glands perform excretory function in prawns, in crustaceans. Clear? Then spiders will have coxal glands. Only insects will have Malpighian tubules. Now, human excretory system consists of kidneys, ureters, urinary bladder, and urethra. Talking about kidneys, see, kidneys will they are situated between last thoracic and third lumbar vertebra. So T12 and L3. Remember it. T12. So where are kidneys present? T12 and L3. This is the level of kidneys. Is it clear? So 120 to 170 gram in weight. So the portion, so the region through which ureters exit or the blood vessels enter, that is called hilum. The broad region is called pelvis, renal pelvis, with projections called calyces. Calyces are the ducts. So the covering of kidney is known as capsule, and overall kidney is divided into outer cortex and inner medulla, and a medulla has medullary pyramids. Is that clear? Projecting into the ducts called calyces. Cortex extends in between the medullary pyramids also. See here. So this whole region is cortex. This is cortex, right? Rest is medulla, and the cortex, you see cortex going in between the medullary pyramids. This forms the renal columns, also known as columns of Bertini. Right now, the unit of kidneys is nephrons, and we have around 1 million of nephrons in each kidney. They are the functional units. Now, each nephron further has two parts. One is the renal tubule and second is the glomerulus. Glomerulus is a tuft of capillaries, while the tubule consists of PCT, DCT and loop of Henle. Now, renal tubule begins with double-walled cup-like structure and the Bowman's capsule. So, renal tubule has Bowman's capsule, PCT, LOH and DCT. Is that clear? Is that clear? Now, this is the structure. You should be able to label it. This is glomerulus. Because of afferent arteriole, the one leaving is efferent. In between the tuft of capillaries is known as glomerulus. Surrounding it, we have Bowman's capsule. So this is PCT. This is the later part is DCT, and this U-shaped structure is called loop of Henle. The part carrying the filtrate downward is descending limb. The carrying the filtrate above is the ascending limb. Is that clear? Now, there are blood capillaries surrounding this loop of Henle, which is known as vasa recta. This is known as vasa. What happened here? This is known as vasa recta. Clear? And many nephrons are going to open in collecting duct. So collecting duct is not a part of one nephron. Now see, Malpighian corpuscle, this term is used for Bowman's capsule plus glomerulus. That is called Malpighian corpuscle or even renal corpuscle. So Bowman's capsule, glomerulus, PCT, DCT, they all lie in the cortical region, while loop of Henle dips into or lies into the medullary region. Now, based on the length of loop of Henle, we have two types of nephrons, cortical nephrons and we have juxtamedullary nephrons. See, in cortical nephrons, we have a very short loop of Henle, while in juxtamedullary, we have a very long loop of Henle which runs deep into the medulla. Is that clear? So this U-shaped vasa recta is it is absent or highly reduced in the cortical nephrons. Talking about the urine formation. First understand, see, urine formation occurs in 3 steps. One is glomerular filtration, second is reabsorption, third is secretion, right? Now, how much blood enters the kidney per minute? That is 1100 to 1200 ml, which is around 1/5 of the total blood pumped out by each ventricle, which is 5000, right? 1/5 of that is 1100. Now, this blood is entering the kidney per minute. So filtration will occur through 3 layers. So just like we have 3 layers in your breathing in your lungs, alveoli, diffusion membrane, right? Similarly, we have 3 layers forming your filtration membrane also. Is that clear? So endothelium of glomerular blood capillaries, endothelium of Bowman's, and the basement membrane. Is that clear? Now, the endothelium of, or the epithelium of Bowman's capsule, they have special cells called podocytes, and the spaces between the podocytes is known as filtration pores or slit pores. It is through these filtration pores or slit pores that filtration will take place. So, almost all components of plasma except proteins pass out into the lumen, and this process is called ultrafiltration or glomerular filtration. So what is the composition of glomerular filtrate? Glomerular filtrate. So glomerular filtrate or ultrafiltrate consists of plasma minus proteins. So everything comes out except for proteins. So overall blood, if we say, so it is blood minus cells minus proteins. Is that clear? So this is the composition of glomerular filtrate. Now, very important, amount of filtrate formed by the kidneys per minute is glomerular filtration rate. That is 125 ml per minute, and if we calculate it for 1 day, it becomes 180 liter per day. Is that clear? Now see, JGA, juxtaglomerular apparatus, is a special sensitive region formed by cellular modifications in the DCT area and the afferent arteriole at the location of their contact. So where DCT and afferent arteriole are close by, near that area the cellular modifications give rise to juxtaglomerular apparatus. Is that clear? What is the significance? This apparatus has certain cells called JGC cells which release renin, and they are going to help in regulating your blood pressure, especially when the blood pressure falls, right? So the urine released is 1.5 liters while the filtrate produced is 180 liters. So this indicates there has to be some reabsorption. So 99% of the total filtrate has to be reabsorbed by the process called reabsorption, which occurs by active as well as passive mechanisms. So actively, glucose, amino acids, sodium are reabsorbed, while nitrogenous wastes and water, they are absorbed passively. Is that clear? Apart from reabsorption, the third step in urine formation is tubular secretion, guys. So the tubular cells secrete substances like H positive, potassium, and ammonia into the filtrate. Is that clear? So you must know where and what is occurring during the urine formation. Now let's see what exactly happens in PCT. So PCT is lined by simple cuboidal brush bordered epithelium. So brush border is going to increase the surface area. So it increases the surface area. So it means maximum reabsorption is occurring at this place. Maximum reabsorption. Nearly all the nutrients, 70 to 80% of the electrolytes and water, they are absorbed here. Is that clear? So maximum sodium, maximum water, glucose 100%, many amino acids, they are all absorbed here in PCT. So it maintains the pH and ionic balance. Is that clear? Now, secretion of H positive and ammonia takes place here, and absorption of bicarbonate ions. Talking about loop of Henle, reabsorption is minimum in the ascending loop. These lines are important as such. Now this is a PYQ line. So the descending limb of loop of Henle is permeable to water and impermeable to electrolyte, while ascending is impermeable to water and allows the transport of electrolytes. Is that clear to all? Is that clear to all? Right? While if we talk about DCT, conditional reabsorption occurs, only facultative if needed. Conditional reabsorption of sodium and water, reabsorption of bicarbonate, is that clear? And secretion of sodium and hydrogen and potassium and ammonia. Is that clear? So all these things all these things are clearly summarized in this one diagram. So you should see in PCT what is being reabsorbed. So arrowhead away from nephron, it indicates the reabsorption. Arrowhead towards nephron shows the secretion, and same is the case here. Now collecting duct, let's talk about collecting duct here. It extends from cortex to the inner parts of medulla. See, we have read the Bowman's capsule, PCT and DCT lies in the cortex while LOH lies in the medulla. But collecting duct runs through the entire cortex as well as medulla. Is that clear? Inner parts of medulla. Large amount of water will be reabsorbed, but here also it will be facultative or conditional reabsorption which will take place. Now, small amount of urea in the medullary interstitium is also required to keep up the osmolarity. Clear? Now, mechanism of concentration of filtrate. Only mammals have this ability to concentrate. So, only mammals have this long loop of Henle and vasa recta. Only mammals have this counter current mechanism. All these points. Increasing osmolarity towards inner interstitium. So, what contributes towards this? It is contributed by sodium ions and urea. Is that clear? Which increases the osmolarity. So what is the osmolarity in the cortex? That is 300. As you go deeper into the medulla, it increases to 1200. That is the maximum. Now gradient is mainly caused by, see, told you, this increasing osmolarity gradient is caused by sodium chloride and ammonia. Is that clear? So together, this special arrangement of Henle's loop and vasa recta forms a counter current mechanism. Clear? Now, this countercurrent mechanism will help in producing this line. Kidneys can produce urine nearly 4 times concentrated than the initial filtrate, because the initial filtrate has maximum osmolarity of 1200, while the urine that comes out, the filtrate, last filtrate, will have around 300. So it decreases to 4 times. Now regulation of kidney function by hypothalamus, JGA, as well as your heart. So see, ADH or vasopressin, who releases it? Neurohypophysis, that is posterior pituitary. ADH facilitates water absorption from later parts of the tubule, preventing diuresis. Increase in body fluid suppresses the ADH release. Are these points clear? Now JGA, juxtaglomerular apparatus, it plays a complex regulatory role. JG, juxtaglomerular cells, release renin. What does renin do? It converts angiotensinogen into angiotensin 1. Now further angiotensin 1 is converted to angiotensin 2. Now this is done by ACE, which comes from your alveolar capillaries, right? Okay, angiotensin 2 is the powerful vasoconstrictor. So what are the actions of what are the effects of angiotensin 2? Number 1, it is a powerful vasoconstrictor. Second, it activates the adrenal cortex to release aldosterone, and aldosterone will then reabsorb sodium. And third, it will also stimulate your posterior pituitary to release more ADH. Is that clear? So 3 things. And in total, all these things working together, renin, angiotensin and aldosterone, they will form a RAS mechanism system. Is that clear? Now, in opposite conditions, so this RAS mechanism is going to function when we have low blood pressure or low GFR. Right? This will stimulate RAS mechanism. But if blood pressure becomes high blood pressure or high GFR, now this will increase the blood flow towards the heart, right? So this will help. Now we need to lower it down. So this will be done by ANF, that is atrial natriuretic factor. This ANF comes from the walls of the atria. So ANF will cause vasodilation, while angiotensin was constricting to increase the heart rate, increase the blood pressure. This ANF is a vasodilator. So the area will increase, pressure will decrease. So we have to work to lower this blood pressure because blood pressure is already high. So ANF is a vasodilator. It decreases the blood pressure and also it is antagonistic to RAS mechanism. Keeps a check on this. Now let's talk about micturition. So micturition is an act of passing out urine. So stored in the bladder till a voluntary signal is given by the central nervous system. Signal is initiated by stretching of the urinary bladder. So this is the first thing. Stretching of urinary bladder. Stretch receptors on the wall send signal to the central nervous system. Central nervous system will initiate the contraction of smooth muscles. It will relax the urethral sphincter, and this in total is known as micturition reflex. And also you see the sequence of steps for micturition. Urine is slightly acidic, pH is 6, and how much urea do you excrete every day? 25 to 30 gram of urea is excreted per day. Now, presence of glycosuria or ketone bodies, that is ketonuria, this is indication of diabetes mellitus. So these are the two things that we check for if we have to check for diabetes. Now role of other organs, like lungs. Lungs will remove carbon dioxide and water. Two things. Liver will remove bile containing substances like bilirubin, biliverdin, cholesterol, some hormones, vitamins and drugs. Is that clear? Then talking about your sweat glands. So your sweat glands, they remove sodium chloride, urea, lactic acid and of course water. And then sebaceous glands will remove sterols, hydrocarbons, waxes through sebum, and even your saliva will also secrete nitrogenous waste. Your saliva contains small amount of urea. Is that clear? Now coming on to the disorders of excretory system. First of all, uremia, excess of urea in blood. So you do carry some urea in your blood, but within a small range. If the level of urea increases beyond that normal range, it is called uremia. Why would that happen? Because if your kidneys are not functioning properly. Now, this is indicative of malfunctioning of kidneys. Now, we can correct uremia by two ways, either by hemodialysis, right, or by organ transplant or by kidney transplant. Is that clear? Now see, how do we perform hemodialysis? Hemodialysis machine is also known as, hemodialyzer is also known as artificial kidney. So first, blood is drained from the convenient artery. This is step number 1. So blood is drained from the convenient artery, right? And second, we add anticoagulant to that. Then it is sent to these cellophane tubes which are surrounded by the dialyzing fluid. Now remember, the dialyzing fluid has the same composition that of plasma except the nitrogenous waste. Is that clear? So once it is sent to the cellophane tubes will allow the passage of molecules based on the concentration gradient. So because it is the blood of a uremic patient, it will have high amount of urea, no urea in the dialyzing fluid, right? So urea from the cellophane membranes, across the cellophane membranes from the blood will pass out into the dialyzing fluid. Is that clear? So after this, now nitrogenous waste are absent in dialyzing fluid. Very important line. Now this blood will be pumped back into the body. The blood from which we have removed excess of urea, this will be pumped back into the body by adding anti-heparin. Is that clear? So that's how we do hemodialysis. So this is the sequence. Kidney transplantation. Once you get a suitable donor, you perform kidney transplantation because hemodialysis cannot go on endlessly. And for that we have to choose a close relative to minimize the chances of rejection. Renal calculi, we call them kidney stones, so made up of insoluble masses of crystallized salts of oxalates. And what is glomerular nephritis? This inflammation of glomerulus. Please tell me these points are clear. So yes guys, we are left with now 3 chapters. One is movement and locomotion. Movement and locomotion here. right? Then we have neural control, and last but not the least, we have chemical control and coordination. Let's start with movement and locomotion. And before we start, guys, all those of you who have joined me now, I want to remind you of this limited time offer. Just for rupee 1, you can get these 25 tests, 10 minus 6 subject wise, 6 major tests, 3 major tests, and 6 overall tests. Now you should start practicing these tests without further delay. Otherwise, this can be really, really dangerous. And please let me know in the chat if all the points are clear. Let's take a 2-minute break, guys. Let's take a 2-minute water break. Right? Give me just 2 minutes and we will all come back in 2 minutes. Class duration, it will take me another hour to complete these 3 chapters. So your next chemistry class is at 8 p.m., right? Today I wanted to take a longer class. Alright guys, the 2-minute water break is over. Let's start with the next chapter again. Let's start with the next chapter, that is locomotion and movement. So see, streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement. In paramecium, cilia helps in locomotion, right? In hydra, we have tentacles for capturing the prey and also used for locomotion. Another important line, all locomotions are movement, but all movements are not locomotion. So the method of locomotion depends performed by animals vary with their habitat and the demand of the situation. Like habitat, for example, in water they use their fins, and for flying they use their wings, and so on. Is that clear? So types of movements, there are 3 main types of movements. One is amoeboid, ciliary, and then we have muscular movement. So amoeboid movement by macrophages and leukocytes, also shown by cytoskeleton elements like microfilaments. Then ciliary movement shown by the internal tubular organs like ciliated epithelium, right, for passage of ova in the female reproductive tract. And then muscular movement by all these organs like limbs, jaws and tongues. They have contractile property. Locomotion requires a perfect, very important line. So locomotion requires a perfect coordinated activity of 3 systems: muscular system, skeletal system, and your neural system. Is it clear? Now talking about muscles, so cilia and flagella are the outgrowths of cell membrane. We have discussed that in previous session when we were discussing the chapter of cell. So flagellar movement helps in the movement of spermatozoa. So sperm shows flagellar movement. Then the canal system in porifera, the conocytes, they also have flagella, right? They also show flagellar movement. Is that clear? Now, what happened? Yes, please. So locomotion of protists like euglena. Euglena also has one long and one short flagella. Trypanosoma, all these show flagellar movement, right? Let's talk about muscle. Muscle is again of mesodermal origin, forms 40 to 50% of our body weight. It has special properties of excitability, contractibility, extensibility, and elasticity. We remember 3 E and C, right? Excitability, extensibility, elasticity, and contractibility. Now based on location, we have 3 types of muscles: skeletal, visceral, and cardiac. Skeletal, also known as striated muscles, they are voluntary and will help in locomotory actions and changing the body postures, right? And then visceral muscles, they are present in inner walls of hollow. Visceral means internal organs, also known as smooth muscles and non-striated, and they are involuntary and they help in, see, transportation of food through digestive tract, gametes through genital tracts. Now moving on to the third type, that is cardiac muscle, present only in heart. They show branching pattern and involuntary in nature. So they have striations like striated muscles, but they are involuntary like involuntary muscles. So nervous system does not control them directly. We have just read in the chapter of heart, right? So heart can be influenced by the medulla oblongata, but it is not directly controlled by heart, right? Now let's talk about the structure of voluntary muscle or skeletal muscle. So skeletal muscle has muscle bundles or fascicles, which all these bundles are covered by a connective tissue called fascia. Now bundles will have fibers. See the structure here. This is the structure of a skeletal muscle. Skeletal muscle will have many bundles. Bundles will have muscle fibers. Fiber is equivalent to the cell. Is that clear? So these are muscle fibers. Now all the terminology that we use for cell will be used for muscle fiber also. For example, muscle fiber lined by plasma membrane, which is known as sarcolemma. The cytoplasm will be known as sarcoplasm. Fiber is now skeletal muscle fiber. Skeletal muscle fiber is multinucleated syncytium. Is it clear? It has many nuclei. So the endoplasmic reticulum will be known as sarcoplasmic reticulum, and it is a storehouse of calcium ions. Now Within this muscle fiber, there are many parallelly arranged myofilaments or myofibrils. Each fibril has alternate dark and light band. Is that clear? Now these light and dark bands, they are formed by two important proteins, actin and myosin. Light band contains actin, also known as the I band or isotropic. Dark band is called the A band or the anisotropic band and contains majorly myosin. Both the proteins are arranged as rod-like structures. Is that clear? It's called myofibrils, and actin filaments are thinner as compared to the myosin filaments. So we call actin filaments as thin filaments and myosin filaments as thick filaments. Are these points clear? Now these filaments will form a special arrangement which will be known as sarcomere. Now see, I band is an elastic fiber called I band. So in the center of the I band is an elastic fiber called Z line which bisects it. So Z line is elastic. The thin filaments firmly attached to the Z line. So the thick filaments in the A band are also held together in the middle of this band. See here. So see this. So this is your Z line. This is your A band. And in the center of A band, we have this M line. Is that clear? A and I bands are arranged alternately. Light band, dark band, light band, dark band, and so on. Is that clear? So functional unit of contraction is called a sarcomere. Now between these two Z lines, this forms a functional unit. So these Z lines, in between the two Z lines is a sarcomere. The central portion of the thick filament not overlapped by thin filament is called the H zone. We have a better type structure here. So this is the Z line. This represents the whole A band. This is the light band. So light band is dissected by the Z line. Is that clear? Now H zone. See, I told you in A band there is both thick filament as well as thin filament. But right in the center, so the central portion of A band where the thin filament is not overlapping the thick filament, this zone is known as the H zone, and this is a PYQ already. Now, structure of contractile proteins. So we have actin filament made up of two F filamentous actin which are helically wound on each other. Each F actin is again a polymer of globular proteins. Then two filaments of tropomyosin are present, right? And a complex protein troponin is present at regular intervals. If we talk about the thick filament made up of myosin, again myosin is a polymerized protein and the monomeric unit is known as meromyosin. So see this. This is your thin filament made up of two F actins, two tropomyosin and troponin at regular intervals. This is your meromyosin. This is a meromyosin. Meromyosin has two important parts. The globular part, head, and the head has arm and head here. This is the globular head, and then it has a short arm and a tail. This is the tail, right? Now the tail forms the light meromyosin, and head and cross arm, this together forms the heavy meromyosin. There are important points, sites present on the head. One is active binding site and second is the ATP binding site. So actin binding site will join with the actin and ATP binding site will hydrolyze ATP to provide energy for the binding. Is that clear? Now, mechanism of contraction, again you have to do that in the sequence format. So first of all, mechanism of muscle contraction is explained by sliding filament theory. Why do we call it sliding filament? Because here these thin and thick filaments are going to slide. Remember, it will be the thin filament that will slide over the thick filament. Thick filament is static. Thick filament is static. Is that clear? Now, muscle contraction. See, here we have the sequence. Muscle contraction is initiated by signal sent by the central nervous system via motor neuron. Is that clear? Second, now this signal reaches the motor unit. This junction between motor neuron and sarcolemma is known as the neuromuscular junction or motor end plate. Now this signal, when reaches the junction, it releases the neurotransmitter. Is that clear? Now after the release of neurotransmitter, it is spread through spread throughout the muscle fiber, and it causes the release of calcium ions. This is a PYQ already. Fourth, these calcium ions bind with the subunit of troponin, and they remove the masking of active sites for myosin, right? Also, the energy is being hydrolyzed for this movement. The myosin head now binds to the exposed site on actin to form a cross bridge. So cross bridge formation is very, very important. Now, as the cross bridge forms, what happens? This pulls the actin filaments towards the center of the A band. Now, inward pulling of these thin filaments, it will lead to shortening of the sarcomere, pulls inwards, and the shortening of sarcomere followed by contraction. So this shortening of muscle or this contraction indicates movement. Is that clear? So these are the stages, these are the sequence. Now, see here. So if we want, the muscle should go back to the relaxed state. So relaxation demands that calcium ions should be pumped back to the sarcoplasmic reticulum or cisternae. This will mask the actin filaments and hence relaxation. Now, if this contraction occurs repeatedly, one after another, repeated activation of muscle will lead to lactic acid formation because of anaerobic breakdown, and this leads to fatigue. Is that clear? Now, muscles have red colored See here, muscles have red colored oxygen storing pigment called myoglobin. Now, there are muscle fibers which have more myoglobin and which have less myoglobin. So, myoglobin is high in red muscle fibers, plenty of mitochondria, so more ATP production, more oxygen, large amount of oxygen, hence they are called aerobic. So, red muscle fibers also known as aerobic. Is that clear? While on the other hand, we have white muscle fibers with less quantity of myoglobin, less amount of mitochondria, less oxygen as well, so they are known as anaerobic. But remember, the white one will have more SER and more calcium, and they are quickly contracting, fast contracting muscle fibers, while these red muscle fibers show slow contraction, and so they go on for the longer time. Is that clear? Now, talking about your skeletal muscle fiber, bone and cartilage, the specialized connective tissue. Bone has very hard matrix with more calcium salts, cartilage slightly pliable matrix with chondroitin salt. Is that clear? 206 bones divided into axial and appendicular skeleton. Let's talk about axial skeleton first. 80 bones, skull, vertebral column, sternum and ribs. If we talk about the skull here, so these all blue highlighted, these are your skull bones, your cranial bones, sorry, this is all skull. These blue lines, these are indicating your cranial bones. So skull is divided into cranium and facial bones. So we have one ethmoid, one sphenoid, one frontal, two parietal, two temporal and one occipital. You should know which ones are paired and which ones are unpaired. While rest of these are showing your facial bones. By the way, we have 8 cranial bones and 14 facial bones. So together they will make 22 skull bones. Is that clear? Now, the single U-shaped bone called hyoid is present at the base of the buccal cavity. Now remember, this hyoid is not considered a part of skull bone. Clear? Then each middle ear will have 3 pairs. Each will have 3 bones. So in total we have 3 pairs, 6 bones in the middle ear. Malleus incus together known as ear ossicles, and they represent the smallest bones, right? Skull region, it articulates with the superior region of the vertebral column. Your skull sits on the backbone, the vertebral column. So superior means upper part. So this is the superior region of your vertebral column where your skull will fit into with the help of 2 occipital condyles. Now because our skull have 2 condyles, that is why our skull is known as dicondylic skull, and we can only have restricted movement till here. If it would be monocondylic skull, we would have been able to move our skull all 360 degree, right? So 360 degree is only possible in monocondylic skull, like in case of birds. Now, coming on to the vertebral column, see here. So the vertebral column, the backbone is made up of 26 bones dorsally placed, right, on the back side. And these 26 bones are known as vertebrae, right? They have a central hollow portion called a neural canal through which your spinal cord is going to run. And vertebral column, these 26 vertebrae are divided into 5 regions. See here, 7 in the cervical region. Now this is a characteristic feature of almost all mammals. Thoracic 12, lumbar 5, sacral 5 bones have fused to form 1 now, and then coccygeal 4 fused to form 1. Is that clear? So this, so 7 in almost all mammals. So what is the function of vertebral column? One, it articulates with your skull. Second, it protects the spinal cord. Also, it provides point of attachment for the ribs and the muscles of the back. Then on the ventral side, guys, we have this central bone which is known as sternum. It is a flat bone, ventral midline of the thorax. Is that clear? Then on the lateral sides, dorsally attached to the vertebral column, ventrally attached to the sternum. Not all of them, but still, right? That is called your ribs. 12 pairs, 24 bones form your ribs. So they are thin, flat bones, dorsally attached to the vertebral column, ventrally to the sternum. Because there are 2 articulation surfaces, they are called bicephalic. First 7 pairs are called the true ribs. See here. First 7 pairs are the true ribs, 8, 9, 10th pair is a false rib, 11th and 12th is a floating rib. Now true ribs dorsally attached to the vertebral column, ventrally attached to the sternum with the help of hyaline cartilage. 8th, 9th, 10th are called false. They are not directly attached to the sternum but join the 7th rib with the help of hyaline cartilage, so called vertebrochondral. Is that clear? And last 2 pairs, 11th and 12th, they are called floating, and they are only connected to the vertebral column, not attached to the sternum or anything on the ventral side. So we call them vertebral ribs also, floating ribs. Is that clear? Then bones of these limbs are attached to the girdles along with the appendicular skeleton. Each limb will have 30 bones, right? So each limb, be it your forelimb or hind limb, will have 30 bones. Let's see here. Yes, please. So this shows your pectoral girdle and the upper arm. Do you see this is your humerus, radius, ulna, carpus, metacarpus, phalanges. These are the 30 bones present in your forelimb. You have 1 humerus, 1 radius, 1 ulna, 5 carpal. Sorry, you have how many carpals? You have 8 carpals, and yes, you have 5 metacarpals and 14 phalanges. Right? Now humerus, the head of humerus fits into the glenoid cavity. Very important. And glenoid cavity is formed by the scapula. So this scapula and clavicle, they constitute your pectoral girdle, the shoulder girdle. 2 on this, 2 on this. So 4 bones form your pectoral girdle. And the glenoid cavity is a depressed area of scapula. So this is glenoid cavity. This is the head of the humerus which it fits and forming ball and socket joint. So is that clear? Now coming on to your lower limb. So lower limb again has 30 bones. So the lower limb has femur, patella, tibia, fibula, then tarsals, metatarsals and phalanges. See, the difference is we don't have a separate bone in the elbow, but there is a separate kneecap, patella. Now, carpals have 8 bones, your tarsals will have 7 bones. Is that clear? Now see, the head of the femur, it will fit into the hip girdle. Now each hip girdle, guys, is formed of, each pelvic girdle consists of 2 coxal bones, and each coxal bone is further formed by the fusion of these 3 bones, ilium, ischium and pubis. And the point of a fusion, point where all the 3 bones fuse, that point of fusion is known as acetabulum. Is that clear? And it is at this acetabulum that head of the humerus will fit into. So humerus fits into the glenoid cavity, femur fits into the acetabulum. Is that clear? Now, are these all points clear? So the 2 halves of the pelvic girdle meet ventrally to form pubic symphysis containing fibrous cartilage, forming fibrous cartilage. So this will be partially movable joint. Now joints, very, very important. See, joints act as fulcrum. 3 types of joints: fibrous joint, cartilaginous joint and synovial joint. See, the fibrous joint do not allow any movement. Example, flat skull bones. And these fibrous joints, they contain dense fibrous connective tissue. That is why they don't allow any movement. They are present between the skull bones known as sutures to form the cranium. Is it clear? Now, in the cartilaginous joint, it is a joint between the two adjacent vertebra. That is one example, and it allows limited movement. And the second example of cartilaginous joint is this, pubic symphysis. Also, told you, containing fibrous cartilage, so allows limited movement. And then the maximum movement is allowed by your synovial joints filled with synovial fluid, considerable a movement. See the examples, very, very important. Ball and socket, humerus and pectoral. Similarly, acetabulum and your femur. Hinge joint, knee joint and your elbow joint. Pivot joint is atlas and axis joint. Gliding joint is between carpals and even between the tarsals. Saddle joint is this joint, carpal and metacarpal of your thumb. I have already discussed these questions in special classes. Now, disorders of muscular and skeletal system. See, myasthenia gravis is autoimmune. Muscular dystrophy is a genetic disorder. In myasthenia gravis, the acetylcholine receptors on neuromuscular junction, they are damaged, causing fatigue, weakening and paralysis. Muscular dystrophy, dystrophin named protein is not formed. It is a genetic disorder. Progressive degeneration of skeletal muscle. Tetani, rapid spasms due to low calcium. Arthritis, inflammation of joint. Osteoporosis is decreased bone mass, hence increased chances of fractures. It is an age-related disorder and also more common in females because of decreased level of estrogen after menopause. And then gout, inflammation of joints due to accumulation of uric acid crystals. We also call it gouty arthritis. So guys, with this we complete movement and locomotion. Please let me know that these points are clear to you all. Yes, please, quickly. Are we done? Let's start with the last second chapter here, that is neural control and coordination. Yes. So this neural control is required to maintain homeostasis. Not only neural control, both neural and chemical control, they are important to maintain homeostasis, right? To integrate and coordinate all the activities in the synchronized manner. The neural system provides network of point-to-point connections for quick coordination, while endocrine will act through hormones. The action will be slower, but it can be longer lasting. Is that clear? Now, talking about neural system, it consists of highly specialized cells called neurons. And in lower invertebrates, the neural organization is simple. For example, in Hydra, while in higher organisms, it is better organized. A brain is present along with the ganglia and neural tissues. Now, the human neural system comprises of central nervous system and peripheral nervous system. Central nervous system has brain and spinal cord. Peripheral nervous system has afferent and efferent nerve fibers. Afferent nerve fibers starting from tissue and organ to central nervous system, and efferent fibers start from CNS to the tissues, right? Now, PNS is further divided in two categories, somatic and autonomic. So, somatic is between your central nervous system connecting to the skeletal muscle fibers, that is your somatic, and autonomic is between central nervous system and involuntary organs. Is that clear? Now, autonomic is further divided as sympathetic and parasympathetic. So, sympathetic works during emergency conditions and parasympathetic during the normal conditions. Now, apart from this, we have a separate classification, part of peripheral nervous system, which comprises of nerves, fibers, ganglia, all the plexus from CNS to the viscera, that is the internal organs, or from viscera to CNS. This is called the visceral nervous system. Now, neuron, as we know, is the structural and functional unit. Each neuron has cell body, dendrites and axon. Cell body has special granular bodies called Nissl granules. Now, Nissl granules are not only in cell body, they are also present in your dendrites, by the way. Is that clear? See. So, Nissl granules and are called dendrites. So, Nissl granules will be present in dendrites as well as in cell body. Is that clear? All right. Now, the axon, see, these fibers contain these fibers transmit impulses towards the cell body. We are talking about dendrites. Now, there is a bulb-like structure called synaptic knob at the end of the axon, and they contain synaptic vesicles. Synaptic vesicles further contain neurotransmitters. Now, based on the number of dendrites and axon present in a neuron, we have three different types. We have multipolar. Now, this represents a multipolar. One axon and multiple dendrites. This is multipolar. Then we have bipolar, one axon and one dendrite, just two processes, and this is found in the retina. And unipolar, uni means only one process, and this one process will be one axon. So our embryonic neurons are usually unipolar in structure. Now, based on the presence of myelin sheath, the axons can be myelinated and non-myelinated. This myelin sheath is always formed by the Schwann cells. Now, at certain locations, myelin sheath is absent, which is known as the nodes of Ranvier. So look at the diagram, how they have represented. Not a very good diagram, but we'll have to use this for labeling. So this outer cell, this pink cell, represents the Schwann cell, right? And the location where this Schwann cell is absent, that is called, this point is called the node of Ranvier. Now, unmyelinated neurons are also enclosed by Schwann cell. So please understand, Schwann cell will be present in either condition, either myelinated, both myelinated as well as non-myelinated. It is actually the placement of Schwann cell that matters, right? Now, unmyelinated are commonly found in autonomous and somatic nervous system. Are these points clear? Then, nerve impulse generation and conduction. So neurons are excitable cells because the membranes are in the polarized state. Means there are two poles. Outer is negative and outer is positive, inner is negative. That is polarized. So channels are present which are selectively permeable to different ions. So this polarized membrane is called the resting membrane, right, where it is more permeable to potassium ion and nearly impermeable to sodium ion. So we are talking about what? We are talking about the resting membrane, polarized membrane. Now, it is impermeable to negatively charged proteins present in the exoplasm. Exoplasm inside the axon has negatively charged proteins and high concentration of potassium and low concentration of sodium. While outside the axon, there is low concentration of potassium and high concentration of sodium. This is all about the resting membrane or the polarized membrane, right? Now, ionic gradients across the resting membrane are maintained by, who is going to maintain this differential distribution? Maintained by active transport of ions by sodium potassium pump. So what does the sodium potassium pump do? It takes out 3 sodium and brings it to potassium at the expense of 1 ATP molecule. Is that clear? Now, see here. So the resting membrane potential is around -70 millivolts. So this potential difference is called resting potential. And what is the value? It is -70 millivolt. Now, when a stimulus arrives, it becomes freely permeable to sodium, right? The membrane becomes freely permeable to sodium. Rapid influx of sodium occurs and this reverses the polarity. Outer surface becomes negatively charged, inner surface becomes positively charged. See, in the resting membrane, outside it is positive, inside it is negative, right? But now the inside will become positive and outside will become negative. Is that clear? So yes, and this polarity of a membrane is reversed and hence it is known as depolarized. Now this first depolarization is called the setting up of action potential, also known as nerve impulse. So what is impulse here? Is that clear? So the distribution of ions is going to change. So the polarity is going to change. Resting membrane is negative on the inside and this depolarized membrane, this is positive in the inside. So how much positive? Plus 30 millivolt. Is that clear? Now we are talking about the inner side always. See, so now when this depolarization, one part of the neuron which has become depolarized, it flows to the next part. This is called impulse conduction. So impulse generated at one side arrives at side B, impulse is conducted. So it is a stimulus induced permeability to sodium is extremely short lived. So the sodium influx which has started occurring here, this is very short-lived. Very quickly, it will be followed by increase in the permeability of potassium. And now potassium will start diffusing out of the membrane. Now this is going to restore the resting membrane potential. Hence, this is called repolarization. Is that clear? So repolarized membrane is very similar to that of the resting membrane. Now, how is it transmitted from one neuron to next neuron? That is called transmission. See, it occurs through special junctions called synapses. Transmission occurs through synapses. So there is a presynaptic membrane, there is a postsynaptic membrane, and in between the two there is synaptic cleft. So synapse is formed by these three components. Is that clear? Now, the synapse can be of two types depending upon whether we require a chemical in between or not. So we have electrical synapse and we have chemical synapse. Let's talk about the electrical synapse. So here, pre and post synaptic neurons are in very close proximity, almost in touch, right? They are in close proximity by the help of gap junctions actually. So this is pre, this is post, and in between there is a channel which is connecting the two. This is called gap junction, right? So basically it's like current is flowing through three wires which are in contact with each other. So the three things will work as one common wire, right? So that is what is happening here. Electric current can flow directly from one neuron to the other. And what brings them closer? It is the gap junction. Gap junction is going to connect the pre and the postsynaptic membrane. Is that clear? Now, electric synapse is always faster than that across a chemical synapse. Is that clear? Now, see here, at the chemical synapse, between the pre and the postsynaptic membrane, there is a synaptic cleft. Now, this synaptic cleft is fluid filled. Is that clear? This space is fluid filled, and in this space, neurotransmitters will be released from the presynaptic membrane. And these neurotransmitters are involved and they will bind to the receptors on the postsynaptic membrane. See here. So this is the presynaptic membrane, this is a synaptic cleft which is fluid filled. These are the neurotransmitters being released and they will attach to the receptors. Receptors are on the postsynaptic membrane. So these three constituents form the synapse. Are these points clear? Now see, neurotransmitters bind to the specific receptors present on the postsynaptic membrane. Now depending upon the neurotransmitter and the potential generated, we can have, the potential can be either excitatory or inhibitory. For example, ACH, that is acetylcholine, adrenaline, they are excitatory, while GABA and serotonin, they are inhibitory. Is that clear? Now talking about your central nervous system, which is the central information processing organ, command and control system, controls the voluntary movements, balance of the body, vital involuntary organs, thermoregulation, hunger, thirst, circadian rhythm, all the activities of several endocrine glands and even human behavior. Apart from this, processing of vision, hearing, speech, memory, intelligence, emotions and thoughts. So in your central nervous system, we have two components, brain and spinal cord. First, let's talk about brain. Well protected by skull and the three cranial meninges, DAP, dura mater, arachnoid, and inner one in contact with the brain tissue is pia mater. Now the brain overall is divided into three parts, forebrain, midbrain and hindbrain. So see, this is the forebrain consisting of cerebrum, so diencephalon, hypothalamus and thalamus, and here we also have olfactory lobes. Now, midbrain is this, and this is the hindbrain consisting of pons, cerebellum and medulla oblongata. Is that clear? Now see the functions. So forebrain consists of these parts: cerebrum, thalamus, hypothalamus. Cerebrum is the major part and is divided into two cerebral hemispheres, longitudinally divided, right? And it is connected by a track of nerve fibers called corpus callosum on the lower end. Is it clear? Now, the outer part of cerebral hemisphere is called cortex, which is highly folded, thrown into prominent folds, right? And these folds are going to increase the surface area for more neurons, right? For holding more neurons. Cortex is the outer area. So it is also known as the gray matter because of the grayish appearance. What contributes to the gray appearance? It is the neuron cell bodies which are concentrated here. Now there are three types of areas present in the cortex area. That is the motor area, sensory area, and the association area. Is that clear? So the large regions which are neither clearly sensory nor motor are called association. Now association areas function for intersensory associations, memory, communication, right? Fibers of the tracts are covered with myelin sheath and they form, now the inner part will form the white matter, sorry. So the inner part, which is made up of myelinated neurons, that will form the white matter. Is that clear? Now the cerebrum, it wraps around a structure called thalamus. Function of thalamus is sensory and motor signaling. Below the thalamus, we have hypothalamus, which is a connecting link between nervous and endocrine tissue. Is that clear? Now, thalamus is a thermoregulator. It controls body temperature, urge of eating, drinking. Also, it has some neurosecretory cells which are going to release the hormones, right? Now, inner parts of cerebral hemispheres have amygdala, hippocampus, and all these structures which form limbic system or limbic lobe, also known as emotional brain, because it controls the regulation of sexual behavior and expression of emotional reactions. Where is it present? Inner parts of cerebral hemisphere. Talking about the midbrain, see, located between the hypothalamus or thalamus of the forebrain and the pons varolai. There is a canal that runs through midbrain which is called cerebral aqueduct, right? Now midbrain has four large swellings, four round swellings on the dorsal side. This is on the dorsal side, here, right? On the dorsal side, which is known as corpora quadrigemina. We also call them optic lobes. Four optic lobes in our brain, two in case of frog. We have to learn about frog also. Is that clear? Now, hindbrain has three components: pons, cerebellum, and medulla. Pons we have read in the chapter of breathing. It has pneumotaxic center, interconnects different parts of the brain and also controls the breathing. Cerebellum, highly convoluted surface, second largest part of the brain. Medulla of the brain connects to the spinal cord, and medulla is involved in controlling the involuntary processes like respiration, cardiovascular, and gastric secretions. Now, very important, brainstem is made up of midbrain, pons varolai, and medulla oblongata. So basically it is midbrain and hindbrain minus a cerebellum. Is that clear? So it forms the connections between brain and spinal cord. Are these points clear? So guys, that was all about nervous control and coordination. And here we are with the last chapter of class 11 and human physiology, which is chemical control and integration. See, I told you neural coordination is fast, point to point coordination, but short-lived. But because nerve fibers do not innervate all the cells of the body. Is that clear? Now, we have endocrine system consisting of these endocrine glands, ductless glands. So they will pour all their secretion in the blood, right? And because blood will go through the whole body, so the target organ can be distantly placed also. Right? Now, what is the definition? Non-nutrient chemicals acting as intercellular messengers. Is that clear? Guys, give me a break. Give me 1 second. Give me 1 second. Yes, guys. All right. So we were talking about hormones, which are non-nutrient chemicals, act as intercellular messengers and produced in small amount. Is that clear? Now, these are the different locations of endocrine glands. And let's discuss the first gland, which is a super master called hypothalamus. Where is it present? At the basal part of diencephalon. Just told you, it is a connecting link. So it has neural role as well as endocrine role. present at the basal part of diencephalon. Diencephalon is made up of thalamus, hypothalamus, and there are other parts also. So forebrain, diencephalon is forebrain. It has several groups of neurosecretory cells called nuclei which produce hormones and regulate the secretion of pituitary hormones, and particularly anterior. Right? There are two types of hormones: releasing hormone, inhibitory. Releasing hormone will stimulate the anterior pituitary, inhibiting hormones will inhibit the anterior pituitary. For example, see, gonadotropin releasing hormone, it will stimulate the pituitary to produce gonadotropin. Then gonadotropin inhibitory hormone, this is term used for gonadotropin, sorry, this is growth hormone inhibitory hormone. Growth hormone inhibitory hormone, also known as somatostatin, it inhibits the release of growth hormone. Is it clear? So two types of hormones. Now these hormones originate in the hypothalamic neuron, pass through axons and release in their nerve endings, and reach the posterior through a portal circulatory system. So how do these hormones from hypothalamus affect the anterior pituitary? So there is a different portal circulation. Like in circulation we have talked about hepatic portal circulation between your intestine and liver. Similarly, again connecting two different organs, that is hypothalamus and anterior pituitary. All right. Now see. Yes. So connecting through hypophyseal portal circulatory system regulate the anterior pituitary. Posterior pituitary is under the direct regulation of hypothalamus. Is that clear? Now talking about what is happening here. Just a second, guys. This is okay. All right. Is that clear? Now, talking about the pituitary gland. Where is pituitary located? In the bony cavity called sella turcica. Now, sella turcica is found in your sphenoid bone, right? Now, it is divided into two regions, adenohypophysis and neurohypophysis. Adenohypophysis is further divided as pars distalis and pars intermedia. Pars distalis is known as anterior pituitary. Is that clear? And it secretes all these stimulating hormones: growth hormone, prolactin, then thyroid stimulating hormone, ACTH, LH and FSH. And pars intermedia secretes melanocyte stimulating hormone. It is almost merged with pars distalis. While neurohypophysis does not secrete, directly produce any hormone. Neurohypophysis, also known as posterior pituitary, it actually stores the hormones produced by hypothalamus, that is oxytocin and vasopressin, right, transported axonally to the neurohypophysis. Now, over secretion of growth hormone leads to gigantism, lower secretion leads to pituitary dwarfism. Middle age, if excess of growth hormone happens in middle age, it leads to acromegaly, serious complications, and can lead to premature death if unchecked. Prolactin helps in the growth of mammary glands and formation of milk in them. Is it clear? Now we know ACTH will affect the adrenal cortex, right? Now LH stimulates the synthesis of androgens, that is testosterone. FSH and androgens together regulate the spermatogenesis. LH causes ovulation in case of females and maintains the corpus luteum. FSH will be important for growth and development of ovarian follicles in females. MSH from the pars intermedia also regulates the pigmentation. Oxytocin acts on smooth muscles and causes vigorous contractions leading to childbirth as well as milk ejection. Vasopressin or ADH we have discussed in RAS mechanism also helps in water reabsorption and electrolytes, reduces the loss of water through urine, causes, and this is called antidiuresis, right? Hence antidiuretic hormone. Now if there is no ADH, deficiency of ADH will lead to excess water loss and dehydration. This is called diabetes insipidus. Is it clear? Now we have a very small gland on the dorsal side. This is pineal gland, dorsal side of forebrain. It releases melatonin, which controls 24-hour diurnal rhythm or circadian rhythm, 24-hour cycle. All the periodic things, events, sleep and wake, body temperature, influences your metabolism, pigmentation, menstrual cycle, and even your defense capability. Then we have thyroid gland. Is that clear? The largest endocrine gland. That is thyroid gland, composed of two lobes. Do we see them? On either side of trachea, connected by a thin flap of connective tissue called isthmus. Now, thyroid has these follicles present and stromal tissue. Follicle will have follicular cells, and follicular cells will make two types of hormones, T3 and T4. T4 will have 4 iodine atoms, tetraiodothyronin, and T3 will have 3. Iodine is essential, remember that. Deficiency of iodine will cause hypothyroidism, and this enlargement of thyroid gland will take place. This is known as goiter. Now, if hypothyroidism occurs during pregnancy, it will impair the development of the baby, leading to defective development and maturation of growing baby, causing stunted growth called cretinism, mental retardation, low intelligence quotient, abnormal skin, even the baby will be deaf and mute. In adult women, it can cause abnormal or irregular menstrual cycle. Now, if cancer of thyroid gland happens, that will lead to hypersecretion of thyroid gland, high levels of thyroxine leading to hyperthyroidism, and then exophthalmic goiter, protrusion of eyeball, and also known as Graves' disease. These are the key words. So what will this thyroid do? See, thyroid hormones, they control metabolism of catabolism, proteins and fats, maintenance of water and electrolyte balance. Another hormone secreted by thyroid gland is TCT, which lowers the blood calcium level, hence hypocalcemic in nature. Then there are 4 glands called parathyroid glands present in the 4 corners on the back side of thyroid gland. They produce a peptide hormone known as parathyroid hormone or PTH. It increases the calcium level in the blood by calcium reabsorption, or increasing the calcium absorption from renal tubules, or increasing the calcium from the from the digested food. So these increase blood calcium, hence hypercalcemic. So both TCT and PTH, they are antagonistic. Now talking about thymus. Thymus is a lobular structure between lungs and sternum on the ventral side, secrete thymosins, cause differentiation of T lymphocytes, which are important for cell-mediated immune system, and also promote production of antibodies, which provide humoral immune system. Now, if thymus is degenerated as you age, so degenerated in old individuals, so decreased production of thymosin and weakened immunity. Is that clear? Now, talking about adrenal gland, see, 2 parts, outer is cortex, inner is medulla, both secrete different hormones. Under production of adrenal cortex hormones, this is called Addison's disease, very important disease. Now medulla, medulla secretes 2 hormones, adrenaline or epinephrine, and the second one is noradrenaline or norepinephrine, collectively known as catecholamines, right? Now these are amino acid derivatives. They respond to stress and hence called the emergency hormones or hormones of fear, fight and flight, 3F hormones. So piloerection means raising of the hair, goose flesh that we call, sweating, alertness, pupillary dilation, right? Increase in the heartbeat, strength of heart contraction, rate of respiration, stimulate and breakdown of glycogen, all is done by this adrenaline. Is that clear? Even the breakdown of lipids and proteins. Now cortex is further differentiated in 3 regions. Outer one is called glomerulosa, zona glomerulosa, middle fasciculata, and inner one is reticularis. All secrete different types of hormones. Is that clear? Okay. Quickly go through and give me 1 second. Right? All right. Now coming on to these three layers. Outer one is glomerulosa, middle is fasciculata, third is reticularis. Now together the hormones from cortex are known as corticoids. See, so outer glomerulosa is going to secrete mineralocorticoids, and fasciculata will secrete these glucocorticoids, reticularis will secrete these sex corticoids. So see, carbohydrate metabolism controlled by glucocorticoids. Cortisol is the main glucocorticoid, regulates the balance of water and mineralites, that is mineralocorticoid, and the main mineralocorticoid is aldosterone, right? Glucocorticoids will stimulate more glucose formation, new glucose formation, gluconeogenesis, break down fat, lipolysis, break down proteins, proteolysis, inhibit cellular uptake and utilization of amino acids, right? Now, the particular one is cortisol, the stress hormone, anti-inflammatory, suppresses the immune response. Now you understand why you get more infections during a stress condition. It stimulates the RBC production. Aldosterone reabsorbs sodium and water and excretion of potassium and phosphate ions. Is it clear? And then this zona reticularis, it releases androgenic steroids in the and helps in growth of axial hair. That axial hair is in the armpits, pubic hair and facial hair during the puberty. Is that clear? Now coming on to pancreas, it is a composite gland. It has both exocrine and endocrine nature. So the endocrine part has aisles of Langerhans. Around 1 to 2 million aisles of Langerhans are present, and they form only 1 to 2% of the total pancreas. So there are two types of cells given here, otherwise there are more. Alpha and beta cells. Alpha cells produce glucagon, beta cells produce insulin. Glucagon is a peptide hormone, acts mainly on the liver cells and stimulates glycogenolysis. So it will increase the sugar level, hence hyperglycemic. Glucagon reduces the cellular glucose uptake. Written. Insulin is again a peptide hormone. It mainly acts on hepatocytes and adipocytes, that means the fat storing cells. It enhances glucose uptake. So the function of glucose is to decrease the blood glucose level, and glucagon is to increase, right? Hence, this is hypoglycemic. So it converts glucose to glycogen. So glycogenesis. So prolonged hyperglycemia leads to complex disorder called diabetes mellitus, and it is indicated by presence of glucose and ketone bodies. Glycosuria and ketonuria. We have read that, right? Now, let's talk about testes. Present in the scrotal sac, have interstitial cells, also known as Leydig cells, and they produce androgens, that is mainly testosterone. What is the function of androgens? Development, maturation and function of male accessory sex organs, like all these ducts as well. Stimulates muscular growth also, facial axillary hair also, aggression, low pitch of voice and spermatogenesis, and also controls the male sexual behavior, and has anabolic effect on protein and carbohydrate. Anabolic, that is why testosterone, these steroids help in muscle buildup, right? Then talking about ovary, it secretes these estrogen, progesterone. Estrogen comes from mainly the ovarian follicles and progesterone from the corpus luteum. Estrogen regulates the secondary sex organs, developing of follicles, appearance of secondary characters, mammary gland development and the sexual behavior, while progesterone supports the pregnancy by helping in the proliferation of uterine membrane. Yes. Then mammary glands and stimulates formation of alveoli and a milk secretion. Now there are other structures also which can secrete hormones, like atrial walls of heart produce ANF, we have discussed. It helps in decreasing the blood pressure by dilation of blood vessels, right? Juxtaglomerular cells produce renin also, and now here they are mentioning about erythropoietin. Erythropoietin helps in RBC production. Then gastrointestinal tract, cells present in the GIT, be it your duodenum or other structures. So 4 peptide hormones: gastrin, secretin, cholecystokinin and GIP. So gastrin acts on gastric glands and stimulates the secretion of HCl and pepsinogen. These are protein digesting. Secretin acts on the exocrine part and stimulates pancreas to secrete the enzymes and water and bicarbonate ions, right? Now CCK acts on both pancreas and gallbladder. So CCK will stimulate the enzyme production. Secretin only bicarbonate and water. Now
CCK, also known as pancreozymin, because it acts on pancreas, stimulates the secretion of pancreatic enzymes and bile juice. GIP, inhibitory peptide. It inhibits the gastric secretion and the growth factors and the motility, sorry. And then there are other non-endocrine tissues which secrete the growth factors for repairing and regeneration. Is that clear? Now, mechanism of hormone action will depend upon the type of the hormone, first of all. So the hormone receptors can be membrane bound or they can be intracellular. See, what will happen? First of all, understand the nature. Both these peptide and amino acid derivative hormones. See, which ones are amino acid? That is catecholamines and peptides. And steroid comes from your gonads and your adrenal cortex. And iodinated ones are just T3 and T4. Now see here, directly come here. Now all the peptide and amino acid derivative hormones, they have these membrane bound receptors, right? So hormone will bind to this membrane bound receptor. Now because the hormone cannot enter, therefore we need a second messenger here, which can be cyclic AMP or calcium ions. Learn the examples here. Now this generation of second messengers will lead to biochemical responses and overall physiological responses. But if we talk about T3, T4, that is iodinated hormones and steroid hormones, here the hormone can itself cross the lipid membrane and enter within the nucleus. So the receptor for these hormones are present within the nucleus, intranuclear, intracellular, right? So directly the hormone receptor complex will be formed. Now because the hormone itself can go into the cell, there is no need for secondary messengers here, right? And then it will lead to some change in the DNA and mRNA, and accordingly it will lead to the physiological changes. Is that clear? So guys, with this we complete NCERT of class 11th. You will be duly notified about the next session on class 12 NCERT. So stay tuned, and I will be practicing all these questions in special classes. And once again, before I leave, I have to remind you about Unacademy All India test series, guys. There are 25 tests in total for just 1 rupee, which you can take at your own convenience. So you have to practice this complete syllabus test. Now, these minor tests are important, which will help you practice, but take complete syllabus test. And also, apart from this, every Wednesday, Friday and Sunday, thrice a week, there is All India mock test series going on, totally free of cost. Please enroll and take these tests and let me know your performance and the challenges that you're facing in these tests. Mere revision will not help until and unless you analyze your performance and know where are your weak points and what to practice on. Okay, on this note, I'll take your leave and I'll see you tomorrow with the next session. Bye guys.